Good morning. Good morning, everybody. Do you hear me well?
Yes.
Yes, great. Thank you. Dear attendees, dear colleagues, dear speakers, I want to welcome you to the second day of this year Biotech Atelier edition. Today's day is dedicated to the satellite event on the topic Innovation in Medicine ATMPs. As you know, Biotech Atelier is organized for the third consecutive year, and this year our event is entirely digital and virtual and better than ever. I want to thank you all for attending the event. I will be your moderator today. My name is Miglena Manova, and I am a Regulatory Affairs Manager at PharmaLex, Bulgaria. I will give some technical insights just before we start. All attendees will be muted. If you have a question you want to ask some of our speakers, you can use the chat box, and our host will forward the question accordingly. Having said all of the above, I don't want to waste more time, and I would like to introduce to you Mrs. Eva Maydell. Mrs. Maydell is a member of the European Parliament. She is the president of the largest organization of associations and civil societies in Europe, the European Movement International. She is vice president of Small and Medium Entrepreneurs of Europe, as well as initiator and co-founder of Education Bulgaria 2030. In the European Parliament, she is a member of the Committee on Industry, Research and Energy, as well as the Committee on Economics and Monetary Affairs. Her priorities are focusing on enhancing skill creation and the use of innovation and technologies, supporting entrepreneurs and investments in Europe. Dear ladies and gentlemen, it's a pleasure for me to present to you Mrs. Maydell.
Good morning. Thank you very much, Miglena, for the kind introduction. I very much want to thank the organizers from PharmaLex for having me invited today, because I think there's a very big difference between me and perhaps all the other speakers in the respective panels and seminars, and that is that I'm not an expert in anything that has to do with medicines. And so I've been very much surprised by the invitation, but on the other hand, I think where we do the link with PharmaLex is on the innovation part and the technological part. So today I will share a little bit on that part. And also particularly, I think we need to say how we met with PharmaLex actually, because it was our common desire to enhance the education in Bulgaria. And so it was on a very, very different topic. And I think if I have to look back 6 years ago when I was elected MEP, it's probably never ever attended anything to do with medicine in terms of seminars or meetings and so on. So it's kind of also my first time to speak at an event that includes that health part. So I'm a regular when it comes to the technology and innovation part, but generally in more tech related sectors. So therefore I'm making in a way this disclaimer because I definitely know way less when it comes to biology and medicine. And so I would not even try to get into that area. And so if there's one word to describe my participation today, for me personally, that's a big challenge. And so that's why I will try to stick to the area that I know better. I'll try to give you a different perspective and an insight on how technology is being regulated in Europe. I know that after me, there's speakers that will go in depth in regulations that you again know better, but I'll try to give you a little bit of an overview where the tech and innovation part stands, at least from my point of view. And perhaps some of the changes that I and some of my colleagues here at the European Parliament aspire to implement. But first of all, I have to share a favorite quote of mine by Bill Gates, because thinking about such an innovative field like science and technology, such as biotech, I can't help it but say that, you know, we very often, and that especially is true for policymakers, I think so, but not only. I think that very often we overestimate the change that will occur in the next 2 years and underestimate the change that will occur in the next 10 years. And I, you know, often with my team, we remind ourselves about the times when, you know, we used to, I mean, this used to be the case for my generation, at least. We used to wait for hours nearby the recorder and the radio to record our favorite song when it came next on the radio. Well, now 10, 15 years ago, you know, we can make our own playlists, and there's ample opportunities of ways to listen to your favorite music. It's a very you know, so to say, one would say, well, that's an easy example, but in a way, it shows you the difference that just 10 years make, even in this part of technology that it is not essential for our survival, let's put it this way, or for our health. So I think one main obstacle of achieving the potential of digitalization across Europe, we call it the digital single market. So we know that Europe's strength lies in its single market, but we understand that when it comes to connectivity, when it comes to infrastructure, when it comes to having the same type of laws for a company to innovate in one country or another, could really position Europe as a world leader. We call that space still a digital single market. So because it's not finalized, there's so much more obstacles to work on. And I really hope that perhaps in a few years' time, if we meet again, we won't call it the digital single market, because the single market will simply be digital. So we would have enabled all this necessarily regulation where that is necessary. And on the other hand, we would have removed the barriers that currently exist in order for innovation to unleash its full potential, in order to position Europe as a world leader in biotech, in robotics, in big data. I think one key thing we have to do is facilitate the dialogue between the tech minds, the scientific minds, and the policy minds, because both innovation and politics will continue in a way to play a role in our societies. But these two elements should get better connected, especially when we see that the whole world is in a way struggling to find, you know, medical, but also social and economic answer to a global pandemic. And this is why I really think we need the business, but also the political leaders to way better understand technology, to be able to see the different opportunities for a positive change in society that technologies can bring. And this is why I think we are also at a very crucial moment when I mentioned global pandemics, and in particularly COVID, is because basically, I think in the past couple of months, so to say, different fields of the economics, in economics that were not necessarily tech geeks, have understood the potential that technology has, not just to connect us through WebEx or Zoom, but most importantly, to change our entire ways of companies, the way they work and the way they produce, and so on and so forth. So that's why sometimes I even find it hard when some colleagues in the European Parliament see it difficult to understand technological and innovative pharmaceutical companies, instead of actually trying to see how we can create a better innovation purpose that could help cure a disease or overcome a social division that we currently face and have in the world today. And so I think we can craft effective regulation only when we eliminate the bias we have towards technology. And unfortunately, that bias currently in Europe is pretty strong. And it's generally, one would say, towards large technological giants. But if you come from an area of expertise, or sometimes even a generation where you do not really grasp technologies, because you have this bias against the tech giants, it kind of transmits in any other type of technology. And so this is a key thing that we need to tackle in order to even start thinking about areas such as biotech with an open mind, and not just already kind of having a shield and putting the shield before us, even before we started, looking at possible legislation. That is only if it's needed. So one thing we did here in the European Parliament is that it's been, as I said, about 6 years that I've been working as an MEP. We have drafted throughout this period different reports, position papers, resolutions on artificial intelligence, for example, as well as data. But none of these have had any regulatory approach. And I would actually argue that that has not necessarily been bad, because in order for you to do regulation that will not stifle innovation, for example, you actually need to understand what you're regulating and what part of it do you want to regulate, so to say. And I think what we finally managed to do, and we've been working on that with a number of colleagues for quite some time, is that we managed to convince the majority of policymakers that if we want Europe to lead in the AI revolution, or on analyzing big data and actually using it, that you could make progress in society, we need a more horizontal and long-term approach on the matter. So just a couple of days ago, we constituted a special committee. So it's a new committee that's going to look on artificial intelligence and technologies in the European Parliament, where for the next year, we'll analyze the future impact that AI has on the economy, as well as investigate the challenges of deploying it. So the idea is to try to, of course, it's a committee where more tech-savvy members sit, but it aims to make the European Parliament the forum and the agora of Europe, where we're going to discuss the newest developments. So not just talk about these things we've been talking for the past 5 or 6 years that are about machine learning bias, or they're about ethical ID, but actually to see how we could use the technologies to boost the progress of Europe forward. And so what we will be doing in the next year is liaising with people such as yourselves from every different area that are pioneers in their fields, in order for us to come with a very cutting-edge report containing factual findings, but also concrete recommendations for common European objectives in the medium, but also long-term, and include major actions needed to reach them. So those actions could be related to investments, those actions could be related to concrete projects, those actions could be related to deregulation, to co-regulation, they could be related to a number of things, but it is the first time that actually the Parliament is working and having a committee that, in a way, you could say it's a little bit forward-looking. Because generally, when you have a special committee, you do a special committee because something bad has happened and you need to investigate it. So now you're kind of like saying, okay, we'll actually do something that I mean, in my opinion, we should have done it 5 years ago, honestly. But anyhow, speaking that we underestimate the changes that would come in 2 years, if I use that Bill Gates quote, you know, perhaps we could say it's better that we have it than not. So I think it's a good chance that we can have and present a more coherent and innovative friendly legal framework for the new tech revolution that could be behind the corner. And I call this approach regulation for innovation. It's a principle that me as a member of this house, I hold very dear, and in all the work that I do and in my amendments of reports and producing reports, I support that principle. Which I always think it has to mean that we need to examine where our regulatory framework falls short and do it with purpose, with target, with a long-term vision, instead of just producing blanket regulation that sometimes end up stifling innovation, or very often produce more red tape for SMEs, or more hurdles, or make it very difficult for SME because they need to put very big investment in order to deliver on that regulation. This is one of the reasons why for the past 6 years I've organized a conference on the topic called Regulation for Innovation, similar to what it's happening today, but on a very more specific matter. And so we've gathered not just the top executive, vice president of the commission and commissioners, but also top executives from tech companies in order for us to basically have a common place where we share our thoughts, search for common solutions on how to adjust our economies to the fast-paced digital age. And this discussion, I have to say, have proven quite fruitful, because it would make us think that, you know, regulations are not just there to impose issues to companies, but to actually unleash their potential where that is possible. So in a way, as I said, you know, maybe today's event is in a way a biotech regulation for innovation, because navigating the complex regulatory environment of advanced therapies means actually that companies need to engage with the regulatory agency early enough, and often throughout the approval process. So if I have to leave you with one word and one message towards the end of my brief opening remarks, that is engage. And I'm sure that's exactly what the companies present today and their representatives do. Be proactive with decision makers, with your ideas, how to overcome regulatory problems. Be also frank and open in those discussions, explain, provide important findings, because I believe this engagement, and as I said in the very beginning, the link between the tech, the science, and the policymakers will become more important than ever in the next years to come. So I look forward to a very interesting and insightful discussion, and thank you once again for inviting someone that really has very little to do with the sector in terms of its final results. Thank you.
Thank you very much, Mrs. Maydell, for sharing your vision on the topic, more in general, like for the technical perspective, but also making this connection to the biotech industry and the pharmaceutical industry. I couldn't agree more with you that actually this very close collaboration between the regulators and the industry is really crucial in order to set the rules in such a way that they do not interfere the development and the progress. That is actually very, very important, and can give us as a society the most benefit of the technologies that we currently have and will further develop. It was the best, and actually we had no doubt that you're the best choice to open this event. So I really want to thank you for accepting this invitation and for sharing your opinion. Thank you. We can proceed now, and for me, in order to set the stage, I have prepared a very short presentation, which thank you for sharing it for me. Can you move to the next slide, please? This will be very short. I will just briefly draft what are ATMPs, different types of them, some characteristics, and I will talk a little bit more about the challenges that actually the ATMP developers are facing during the development process, and I will present you the event agenda and the speakers that accepted the invitation to attend and speak today. Can you please move to the next slide and the next one? So advanced therapy medicinal products are actually a new category of medicines with a very wide therapeutic potential. They represent a breakthrough in the therapies and in the healthcare systems and patients, and they give us great potential to prevent and treat diseases that currently were considered untreatable, like different inherited diseases, cancer, immune diseases, cartilage defect, Parkinson's disease, and many others. Can you please? No, we are speaking here again, sorry. The umbrella term actually of ATMPs consists of 4 distinctive classes of products. These are the somatic cell therapy medicinal products, the tissue engineered products, the gene therapies, and also the combined ATMPs that actually consist of a medical device and a cell or a tissue component. Can you please change to the next slide? ATMP have some very significant characteristics as they originate from living cell and contain viral vectors. They are characterized with a high degree of complexity. Usually the cells are derived from a patient or an allogenic donor and are processed in the laboratory, and later they're administered back to the patient, usually in a hospital environment. Gene therapies are designed to introduce genetic material into living cells in order to compensate some abnormal genes or to enhance the expression of a beneficial protein. With all those characteristics, actually, ATMPs have initiated a shift from the currently traditional one-size-fits-all approach towards the personalized medicinal strategy. Can you please shift to the next slide? And the next one. Thank you. However, the ATMP development field is relatively new still. And despite the fast development of the technologies, there are still many challenges that the ATMP developers actually need to overcome in order to reach the point when they can place the product on the market and actually reach the patients. In order to mark some of the challenges, I will refer to a survey that was conducted among companies in Europe who are developing currently ATMPs. This survey was performed by the Division of Pharmacoepidemiology and Clinical Pharmacology in the Utrecht Institute in the Netherlands. The study actually yielded 243 challenge descriptions, and after they were classified, some very significant domains were marked, and these are the top 3 challenge domains: the regulatory domain, the technical domain, and the scientific domain. After further classification of the domains into sub-teams, 3 teams were mostly mentioned by the ATMP developers. This is dealing with the country requirements and actually meeting them, manufacturing, and trial design. In the survey, there is also one very interesting challenge domain that is not among the top 3 obviously, but I find it very significant, and I will also talk a little bit about it. This is actually the domain of the financial challenges. Can you please check to the next slide? Here I have presented to you in a table format extract of the survey and actually some of the challenges, domains and themes that were identified by the ATMP developers. The ATMP developers that actually took part in the survey are 68, and half of them are located in Western Europe, in UK, Germany, and France. 16 of the companies are primarily based in the United States, but they also develop ATMPs for the European market. Most of the companies develop cell-based medicinal products, followed by genetic medicinal products, and also combined ATMPs. 35 of the respondents, which is 51%, they report developing more than one ATMP, and 10 of them actually work on different ATMP types simultaneously. Together, the 68 respondents in the survey are developing 126 ATMPs. Can you please move to the next slide? This slide actually drafts the most significant challenges with regards to regulatory. And the two main challenges identified are the fact actually the applicant should work towards the European centralized procedure in order to obtain a marketing authorization. And the second one is all the challenges they're facing while compiling the regulatory dossier, which is actually crucial and needed for the marketing authorization. Also, on European level, it was identified that the very positive pre-submission interaction with EMA committees is considered challenging. According to the studies, actually, these developers, they report that this is more extensive and more detailed interaction with the authorities in comparison with developing a non-ATMP product. The regulatory information requests they receive during that discussion usually are found to be difficult to fulfill, and often many additional data is requested during the discussion and the assessment, which actually significantly increases the associated costs. Additionally, I found this very significant, so I'm mentioning it very briefly, that the gene therapy medicinal product developers actually are more challenged by the country-specific requirements, and this is due to the different national regulations regarding the genetically modified organisms. Can you please move to the next slide? Thank you. As my background is mainly regulatory, I couldn't help myself but put one slide regarding the different possibilities of very early interaction with the European agency regarding the ATMPs. And here I will not talk about the Committee of Advanced Therapies, because this will be further discussed in our event today in very much details. But I want to mark the possibility of the developers and applicants to reach out to the Innovation Task Force Committee, which is a multidisciplinary committee established in 2001. The main focus and tasks of this committee is that it can be actually reached in a very early stage in the development process, even prior requesting scientific advice. And the purpose is that Innovation Task Force actually can give advice and guidance in the development process, the classification of the ATMP, the orphan drug designation, and of course even scientific advice. Can you please move to the next slide? Thank you. So the second significant challenge captured in the survey is actually related with the technical domain and the technical challenges. One of the most significant issues is the challenge developers are facing when they need to scale up the process. They face serious inconsistency issues that they report the most. In these cases, when they reach out to seek external help from CMOs, it also turns out to be difficult to engage with the appropriate and the best CMO in a specific case. Additionally, GMP legislation is also interpreted differently across the different member states, so it's a bit challenging to meet the GMP requirements. And national competent authorities also request very often information, additional information on quality standards. One of the biggest challenges in this technical perspective is noted when actually manufacturers and ATMP developers are looking to in-license products originated from academia or hospital research. In these cases, the method of manufacture there turn out to be quite unsuitable for a later stage development. Even though it is, of course, recognized that academia has a very significant expertise and background with regards to science and progressive vision, they also have some background challenges and difficulties regarding the technical aspects that can be further improved. Based on the survey, ATMP developers report that there is sometimes lack of GMP compliance with these products originated from academia, which makes it quite difficult to obtain information regarding key aspects of the ATMP development. Another technical challenge is related with the non-clinical and technology and toxicology programs, who often tend to be extremely abbreviated and consist of just one or two studies that are not always beneficial. Such studies are sometimes not conducted in accordance with good laboratory practice and may be considered insufficient. Supply chain challenges are also mentioned here because it is challenging to supply the product with regards to very short shelf life and different preservation and quality control solutions are needed with these regards. Can you please change to the next slide? Thank you. The third domain is related with scientific challenges. They're related with the fact that ATMPs are currently and most often developed for very rare diseases that haven't been treated before. So this is associated with very specific set of challenges, mostly reported to be the challenge to come up with the trial design. Another significant issue is the very low patient numbers and little knowledge about the disease progression. Also, very different, very little knowledge about the disease progression. And challenges associated with the creation and interpretation of the endpoints of the new indications that are about to be registered. here it is often reported by SMEs that they're facing challenges with the lack of relevant animal models available. Can you please move to the next slide? And we come to the fourth and last domain that I will briefly mark here in this short presentation, the financial challenges. They actually occur because the development cost of the ATMP products is very high. At the same time, ATMP developers and applicants, they face significant reimbursement uncertainty. And combining all of that, of these two factors, we actually see that some of the already registered ATMPs have been withdrawn from the market due to commercial. It is interesting here to be mentioned that actually it is equally mentioned by the participants in the survey that the uncertainty in the reimbursement perspective and actually funding are equally distributed between them and considered equally challenging. With having said all that, I will not go in any more details because I would like to give that opportunity to our speakers who accepted to participate. Can you please move to the next slide? And the next one. So today we will proceed with Violeta Georgieva. She will tell us about the European ATMP development landscape. She is a legal affairs manager at the EuropaBio Association. And now I would like to give the stage to Violeta. Violeta, please.
Good morning. Good morning. I'm just going to try to share my slide. And I need the help of Theodore. Yes, I'm enabled now. Thank you. Hello? Sorry. So I hope you can see my slides now.
Yes, yes.
Just I wanted to start with by saying that it's really a privilege for me to be here with you today and have the opportunity to speak about the development landscape and the outlook of ATMPs in Europe. The goal of my presentation will be that I give you the big picture, and as you've seen, we have a very nice lineup of speakers. So they will zoom in on many of the aspects that I will mention in my presentation. And actually, my personal goal is that by the end of this presentation, I leave you all convinced in the transformation of power of ATMPs in three main domains. So first of all, for the lives of patients and their families. Then secondly, for our healthcare systems. And thirdly, for Europe's competitiveness potential in the field of technology and particularly in the field of life sciences and biotechnology. So first, just a quick word about who I represent and work for. As you've heard from Miglena, this is EuropaBio, the European Association for Bioindustries. We do represent biotech and biopharmaceutical companies at the most innovative end of industry. And what we truly believe in and advocate is that capturing the full potential of biotechnology and life sciences is going to provide the solutions that the world and Europe need for many of the challenges that we face today, starting from health and well-being, aging, to climate change, to zero waste and resource-efficient economy, society, and environment. And just quickly mentioning COVID, actually, the current trying situation with COVID only increased the attention to the sector of biotechnology, as the whole world now is looking at the biotech industry to provide a solution in the form of a vaccine or a treatment. And I'll also specifically tell you a few things about ATMPs in times of COVID as well. So we truly believe that biotechnology is health. And just to illustrate that, you may be curious to know that there is a 72% likelihood that the next treatment for cancer or a rare disease will come from an emerging biotechnology company. We're very proud actually of our more than 40 healthcare company members because they spend on a yearly basis more than around 70 billion euros in R&D, and more than 50% of those companies are active in the space of ATMPs. And these are startup companies through mid-sized companies to bigger biopharmaceutical companies. So now on ATMP specifically, so you've already heard from Miglena what they are. I give you on this slide what you can read, or the definition that you can read on the website of the European Medicines Agency. But what I really truly want you to take away with you is that these medicines are living medicines. So they are with the patient to stay, and some of them they change specific aspects of the human DNA. This is very specific and this is very powerful. This is really bioscience in its prime, and it is very exciting. We're living in very exciting times because for the first time probably ever in human history and in the history of medicine, biotechnology is actually delivering solutions to the most challenging of conditions. So I'm really truly excited to be in this field. Here on the next slide, there is just a list, a non-exhaustive list, I should say, of the possible applications of ATMP. So here I've just tried to give you some of the very well-known conditions which we see still as very challenging. So these are various forms of cancers, also diseases, neurological diseases, many rare diseases. And now with ATMPs, we have truly the opportunity to address those in a sustainable way. And also in this aspect, I wanted to briefly mention this newer category of ATMPs, the gene-edited ATMP. So think of CRISPR, because actually earlier this year, it was June, I believe, when some more clinical data about their clinical efficacy was published, and then we saw a data about the CRISPR therapy against sickle cell disease, and it showed that actually 9 months post-treatment, the patients are transfusion-free, which is really truly remarkable. And one of the big questions for the ATMP sector now is whether advanced therapies will be able to provide solutions to chronic diseases, so long-term conditions. And this is the ultimate goal of the ATMP sector, to be able to treat diseases like age-related blindness or Parkinson's or diabetes. So a lot of potential here, truly. On to the ATMP specificities now, and Miglena has already given you some of those. But what I wanted really to say is that they truly open new cure horizons now, because they address very often the disease at its genetic root cause, and you can treat the patient once. So you need one intervention, and that could potentially be a cure for life. This is astonishing, really. And so we are talking here about, as Miglena also pointed out, about tailored medicine. These are very often bespoke treatments. And because they're so revolutionary, we also need policy and regulation. And I very much like the comment made by Mrs. Maydell that we have to really overcome the technology bias. So we need regulation which is equally revolutionary, I believe. Because these therapies, they are very complex to deliver and to manufacture, as also Miglena pointed out. Just to illustrate what I have in mind is that you may have heard that to deliver a CAR T treatment to, let's say, a blood cancer patient, you usually need the involvement of about 150 doctors, nurses, scientists, couriers, and these people are sitting very often geographically in very many different places. So honestly, this is the most complex supply chain in the history of medicine. And this is why these therapies are also coming with very high upfront costs. So we have to address, we have to adapt our systems of reimbursement, for example, and if you stay with us this afternoon, you'll hear more about it from the speakers on the panel later today. what should be also noted is that ATMPs will make up a bigger and bigger chunk of the biotechnology and biopharmaceutical sector, which also means we have to prepare for that. In Europe specifically, there is a lot to do here because we have to make sure that we remain competitive, and we are truly focused on coming up with technology-friendly and enabling regulation and policy solutions. And now I wanted to give you actually some numbers, and I'll present first the global landscape of ATMPs. The data that you see on the slide capture the situation as of end of June this year. And despite COVID and despite the fact that for many people 2020 will be remembered as the year that probably wasn't ever, this was a very remarkable, this has been so far a very remarkable year for the ATMP sector, which showed also significant resilience to all the turmoil. For the first time, we see more than 1,000 ongoing clinical trials with ATMPs worldwide. And I believe just over 400 of them. We see more than 1,000 companies active in the field of ATMPs, and more than 400 of them are in clinical stage of development. And as you see from the map on the slide, the US and North America altogether, they remain a leader in this sector, with more than half of the companies headquartered in North America. Europe is still making up a quarter of the sector, again, in terms of the number of companies headquartered in Europe. But we also see that it's starting to lag behind. So there are many issues to address in Europe specifically. And now onto the European landscape. So if you zoom in on the European map, you can see the clear leaders, and I believe they were mentioned by Miglena and the respondents to the study that she presented. So these are clearly the United Kingdom, Germany, France, and Israel. As of end of June 2020, there has been some positive news on the regulatory side in Europe, and that was the approval of the first gene therapy against spinal muscular dystrophy, Zolgensma. And now the first patients are close to benefiting from this remarkable therapy. I move on now to the numbers of clinical trials and financing in the ATMP sector. Again, the data is from end of June 2020. So you can see clearly the resilience of the ATMP sector despite COVID is showing here very clearly. More than 1,000 clinical trials worldwide. Again, a quarter of them have been sponsored by a European or Israeli developer, 270. And because you've mentioned, Miglena, also financing as one of the challenges for the sector is truly so. But again, despite COVID, the ATMP sector and to 2020 are actually set to show a record-breaking year in the total of financings. For the moment, we're at 9 billion at global scale, which is already more than the total of the entire 2019. And if you also compare to 2018, which was the previous best year in terms of financing, therapeutic developers have already raised 80% of the full year total of 2018. So we expect 2020 to be really a very, very good year, even the best probably in the history of ATMPs. The European and Israeli financings look also very good. We're at 2.3 billion so far as of June 2020, and this has already doubled the numbers of 2019. So you see clearly an increase here, and we are very happy to see these numbers. And that's my favorite slide. It's the patient impact of ATMPs. And here, unfortunately, I won't have the time to stop on each and every of these therapies on the list. And again, this is just a selection of a few of the most recent ATMPs. I'll probably want to focus on the last two because they're approaching now the patient access stage. So the one before the last on the list is a very new gene therapy against hemophilia. And the long-term data is showing, again, astonishing results at 95% reduction in bleedings, so very good efficacy. Zolgensma, which, as I said, was approved in the EU in May of this year. Again, the data from the longer-term study shows that all 10 patients were kept alive, and they're also free of permanent ventilation. So a lot of promise here, a lot of potential. And what is even better to hear and to tell you today is that actually both the European Medicines Agency and the Fed and the FDA in the US expect a wave of applications and authorizations of ATMPs in the years up to 2025. The EMA specifically only by the end of next year expects around 20 new applications, and most of them are gene therapy. I remember yesterday the chair of the CAT, she said 80% are going to be gene therapy. So as I work in EU affairs, I wanted to give you a snapshot of the political and regulatory ambitions across the EU. And I wanted to focus here on three main documents that we have seen or are expecting to see this year. First, I'll start with the EMA Regulatory Strategy to 2025. The document was adopted around March of this year. And as you see, one of the strategic goals of the EMA will be to actually support the integration of science and technology for medicines development. And under this objective, they want to specifically support the translation of ATMPs into patient treatments. Some examples of the actions that they want to undertake, you see on the slides, but basically these are, they want to enhance the scientific advice, they want to support evidence generation, they also want to cooperate better globally so that they can back regulatory conversions. So we have very high hopes at EuropaBio, and we are really looking to see whether these actions will be followed up in an appropriate manner. The next on to another flagship initiative of the European Union. That's the Europe's Beating Cancer Plan. We expect to see this document by the end of this year. And why I'm mentioning is because the European Commission has clearly said that they look at cell and gene therapies as a very transformative approach to cancer treatment, and they also do appreciate all the specificities of ATMPs, and they also do recognize the need to adapt our healthcare systems model. So again, a lot of hope here. And then in terms of the promise of ATMPs for cancer, you may know that cell therapies are actually best represented in oncology. And actually blood cancers such as leukemia and lymphoma were the first breakthrough domains for cell therapies. I gave you also some numbers here. It's more than 60% of ATMPs are developed in oncology altogether, and about, I think, 64% of all clinical trials with gene therapies for rare disease are for rare cancers. So I believe there is a lot of hope here for patients who are at a very critical juncture. So we believe ATMPs, and we hope ATMPs will be able to respond to those patient needs. And finally, I wanted to give you a word on the new EU pharmaceutical strategy, which will be adopted around November of this year. Again, a lot of focus on gene and cell therapies because this is the emerging science in medicine. And one of the primary objectives of this strategy, the European Commission is saying, is going to be to improve access, availability, and affordability of advanced therapies. So again, we're looking forward to seeing a lot of action in these domains. And at the bottom of this slide, you also see one of the questions from the public consultation of the European Commission on the pharmaceutical strategy, which tells you that there is also an increased attention to the hospital exemption, the so-called hospital exemption for cell therapy specifically. And I will not address it because I know there will be a specific discussion at the Biotech Atelier on this point, but wanted to highlight that fact. Yes, and COVID. No presentation today can end, I believe, without mentioning COVID at least a few times, and I actually decided to dedicate a full slide on COVID because I thought you may be curious to know that there are actually 11 ongoing clinical trials with ATMPs targeting COVID. And there are different approaches. Some of those ATMPs are developed to promote an immune response. Others aim to block inflammation, for example, lung inflammation, which is one of the complications in the serious cases of COVID. Or they also aim, some of the research projects, they also aim to repair tissues damaged by, again, COVID implications. Another curious fact is that some of the very promising vaccine candidates are based on research with gene therapies. So again, we are very hopeful that ATMPs could actually be part of the solution to the current situation with COVID. On the very positive side, we have seen, and again, going back to the point about the bias towards technology, we have seen a very positive precedent in the EU. It was, I believe, July when the European Commission and then afterwards the European Parliament and Council supported a derogation from the GMO requirements for clinical trials, which also Miglena addressed in her presentation is one of the main obstacles. And we at EuropaBio look at this as certainly very positive development, and we hope it will be built up upon to streamline the process for clinical trials with gene therapies containing or consisting of GMOs. On the less positive side, COVID did have an impact, a rather negative impact on specifically the planning and conduct of clinical trials with ATMPs, also on data collection and so on. I looked at some recent data which were published 2 weeks ago. And they evaluated that the number of new subjects entering clinical trials as of September 2020 has decreased by 30% compared to the pre-pandemic levels. But this is still, so the trend is sort of reversing now, because actually this third quarter is much better than the numbers we saw back in April, when the numbers of subjects entering clinical trials decreased by 70%. So again, I believe we should remain optimistic. The sector is very resilient, and we hope it will continue to show the results that everybody looks forward to seeing. And off to my conclusions now. Again, a very strong sector despite the pandemics. We have seen we are seeing remarkable numbers of clinical trials and investment so far in 2020 in the ATMP sector. 2020 is going to be a record-breaking year for financing. So at least on this point, we can, I think, be a bit relaxed for the moment. What we can say also with certainty is that there will be a wave of ATMPs reaching patients, payers, and regulators in the next 3 to 5 years, and this is why we have to prepare. Europe, unfortunately, we see is lagging behind in terms of clinical development. I like statistics and I like to illustrate, but you should know that between 2014 and 2019, the number of clinical trials with ATMPs in Europe were less than half of the numbers that we see in the United States and Asia. And also the trend of increasing those numbers is also far, far away from these two other geographies. So we really need to set our ambitions right in this sector, and we need to prepare and transform our healthcare systems to make sure that patients benefit from these revolutionary treatments. And with this, I really want to thank you for your attention and the time I was given. And I hope we can have a fruitful discussion after the presentations. Thank you.
Thank you very much, Violeta, for giving us this very detailed and interesting overview of the ATMP landscape. You gave very interesting and useful information to the audience, but I will take with me the message that despite COVID, this year is the best with regards to ATMP development. So thank you for that positive message in this global pandemic situation that we are currently now in all together. So I will give now the stage to our next speaker. Zaklina from PharmaLex Germany will give us an overview of the ATMP regulatory strategy. Zaklina is Director Innovative Therapies in PharmaLex, and it is a pleasure to me to introduce to you Zaklina.
Yes, so hello. Thank you very much, Miglena, for your kind introduction. And thanks also to the previous speakers. And well, Eva, you yourself and Violeta, I think really you made a very good, you made very good points with what you provided already to us and set the scene. So we are kind of going positive into the next presentations. And so as I'm a regulator, so working at PharmaLex and already more than 10 years with ATMPs now, my talk today is about well, the ATMP framework. I will shortly also talk about the definitions once more. If you're very accustomed to that, you can, of course, get your coffee, but be back in 10 minutes because then we are going on with strategy. No, that was the wrong one. Here. So we have a first part on the regulatory overview, and we'll also give some updates on what's happening in the EU. And then the second part is about regulatory strategy. We will look at specific considerations, incentives in the EU and US. I will also touch a little bit Japan and the authorized products, as well as the incentives and how they can be used. So if just looking at the European situation to give an overall view of the framework, I mean, of course, we have the Medicinal Products Directive, which is covering all medicinal products. And then we have the regulation 726/2004, which is defining in the annex which products are authorized via the single centralized European procedure. And in this annex, we also find the ATMPs, and the specific ATMP regulation, as you know, is the regulation 1394/2007. And then we also have a framework on the starting materials of blood products and human tissues, and some more directives which are shown here. And in case of combination products, we also might have to consider the medical device regulation. There are three groups that has been mentioned before, but I want to go a little bit more into detail into these groups now. So the first two groups, the gene therapy and the somatic cell therapy products, have been defined already for quite some time in the annex of the directive. But so the tissue engineered products were still kind of missing when the regulation came into place. So that was it came into place 2008, and so also the tissue engineered products are now included. Sometimes we use the word cell-based medicinal products in Europe for somatic and tissue engineered products. So we'll start with the gene therapy products. So according to the definition, they contain of recombinant nucleic acids used and administered to human beings, and they should be regulating, repairing, replacing, adding, or deleting a genetic sequence. So the therapeutic effect then relates to this recombinant acid and or to the product of that genetic expression. So very often that is an enzyme then. What is important to mention is that gene therapy medicinal products do not include vaccines against infectious diseases. So, Violeta touched what's happening on the COVID-19 in the area, and you all know about some of our frontliners, mRNA vaccine productions, which are vaccines against an infectious disease, so they're not included in this definition. Okay, what types are there? So I like this picture on the right from the National Library of Medicine. It's very nice showing what gene therapies are. So if you see this small nucleic acid sequence in the packed on the left-hand side, and when that is included into the human cell and makes or replaces the gene, so that is the gene therapy. And there can be there can be naked, like plasmid DNA. There can be the nucleic acid can be included into a vector, which might be a viral one, or also, for example, a bacterial vector. And then we also have, of course, the genetically modified cells. And you can say that depending on whether this the human cell is modified by the vector, including the nucleic acid inside or outside the human body, you speak of ex vivo or in vivo gene therapies. So examples for ex vivo gene therapies are CAR T cells. So they're, for example, just Yescarta Cymria. We have also Tecartus in the US right now. Then for in vivo, for example, Imlugic as an oncolytic virus therapy, or Zolgensma, and we have many more. So coming to the next group, somatic cell therapy medicinal products. So there we speak of substantial manipulation, and the tissues should also, or as an alternative, not be intended to be used in the same essential function. So that means that, for example, here, if you have bone marrow-derived progenitor cells and use them for treatment of myocardial infarction, it would be a different function than in the beginning, a different intended use. And then as a second, of course, these drugs have to treat, prevent, diagnose a disease based on a pharmacological, immunological, metabolic action. Okay, and then coming to the tissue engineered products, so they consist of engineered cells or tissues, and these again are also defined as either substantially manipulated or not used in the same essential function. So we have two examples again. So substantially manipulated, you would consider chondrocytes for repairing cartilage defects in the knee, and for the same indication for the cartilage defect, if you take mesenchymal stem cells, they might also they will also be considered a tissue engineered product because it's a repairing function, but it's a different function than the original one of the stem cells. Okay, and then the difference to the somatic stem cells then is that these tissue engineered products are regenerating, repairing, or replacing human tissue. The cells can be combined with other materials, and they can partly also be non-viable. Here are some examples with some pictures. So for the three groups on the left side, you see the tissue engineered group. So there are two authorized with the EMA now, spheroids to treat cartilage defects in the knee, and Holoclar, so an orphan medicinal product for treatment of limbal stem cell deficiency, and 2 examples for somatic stem cell therapy, Alofisel, which are stem cells derived from fat tissue to treat morbus Crohn anal fistulas. Then we have Provenge, dendritic cells to treat prostate cancer. We'll see, and unfortunately that product is not on the market in Europe any longer. So then we have, for the gene therapies, 2 examples. This is Luxturna, an adeno-associated virus to treat a specific genetic eye disease, and the already mentioned CAR T cells, and for example also Zynteglo to treat or cure beta thalassemia. So those who heard yesterday's talk from Martina Schüssler-Lenz, of course, also had a very nice overview again on the ATMP landscape. And we also heard before about the possibilities for ATMPs. So, and if you're not sure about the classification, you can also ask for a recommendation of the Committee of Advanced Therapy for classification. And you can also consider this already as a part of a strategy, regulatory strategy, because in some way you can influence these classifications if you see a need to do so, of course, depending on your mode of action. As I said, that we would not talk only about Europe, but also look a little bit further to other regions, especially the US. So I just depicted here the US definitions. Just take some highlights. So we have 2 groups, basically the human tissue the human gene therapy and the somatic therapy, which both have to be authorized via BLAs. And then the human gene therapy, that's quite similar to what we see in Europe. The material, the cells need to modify, manipulate the expression of the gene, alter biological properties of living cells for a specific therapeutic purpose, and lead to a sustained effect. This can be for prevention or treatment or cure of a disease in humans. The exclusions also are mentioning the prophylaxis of infections and diseases which are excluded, but they also mention the treatment. So that might be a little bit different to Europe than with respect to infectious diseases. Yeah, and then for the somatic cell therapy group, this is a little bit broader than we see this in Europe. So it's including allogenic, autologous, or even xenogenic cells that have been propagated, etc., to alter the biological characteristics. You have seen this similarly in the European definition. Examples are lymphocyte therapy, but also cultured cartilage cells, so what we would consider as tissue engineered, and also unrelated allogeneic hematopoietic stem cells. And I just want to mention here that also in the European context, unrelated allogeneic hematopoietic stem cells used for transplantation purposes, for example, in cancers, are not considered ATMPs, but are regulated under the tissue directives. Okay, so and then there are also the inclusions of minimally manipulated human cells and tissues. We have heard already a bit about strengthening the landscapes of agencies. So the FDA goes on supporting the innovation, especially of gene therapy products. 2017, we had this program of the European Commission and the medicines agencies on an action plan on ATMPs. I give a very short update on that. And we just heard from Violeta also about the very recent EMEA regulatory science plan to 2025. So there's something happening. So within this action plan that I mentioned, this is a document which listed several guidances which needed to be updated or which didn't exist so far, and some other topics. And here are the guidances that have been updated and created so far. So a very important one certainly is the one questions and answers papers on comparability. Then we have also some procedural advice on ATMPs. So based on the experience the CAT has collected during the last years, also including more flexibility on clock stops and things like that. Then we have the guideline on quality, non-clinical, clinical aspects of gene therapies, where also gene editing is included. Then certainly important GMO aspects on investigation medicine products. There's also, Commission trying to a bit more harmonize the European landscape. And we also have the parallel consultation with regulators and health technology bodies. There is a nice leaflet for developers, so maybe more for early developers, researchers, to get an overview over all the different incentives we have in Europe. And with respect to GXP, there's also some updates. Of course, on the right-hand side, you will all know we have by now the guideline of GMP guideline for ATMPs. We also have GLP principles in relation to ATMPs already since 2017. And then there are also updates on the GMDP working group with respect to exemption of batch controls and out of specification batches. Yeah, some ongoing guidelines, so safety and efficacy follow-up, the guideline on genetically modified cells, and also the investigational advanced therapies. So if you can also have a look already in these guidelines, especially the later one gives you some interesting considerations. Okay, we come to the strategy. So first, I want to give an overview of the special considerations, incentives we have in Europe and the US. So both regions have some kind of orphan medicinal products designations with relatively similar definitions. The US also has this incentive called Rare Pediatric Disease Voucher. Then with respect to development pathways, we have PRIME now. We had in earlier years adaptive pathways and had a lot of learning from that, including the parallel advices with the HTA bodies. Then in US, we have fast track breakthrough for already quite some time. And since I think it's 2016 or 17, the regulatory medicines therapy designation, ARMAT designation in the USA exists as well. With respect to the legal framework, both regions have a similar concept, conditional marketing authorization, respectively accelerated approval. As a first step, marketing authorization based on more preliminary clinical data to be updated later. And we also have in Europe the exceptional circumstances authorization in the case where there's not enough data. For example, if you're a very rare disease, have a rare disease product. We had this, for example, in the case of Glybera. Then the review times for marketing authorizations can also be shortened in certain cases. So we speak of accelerated assessment in the Europe and priority review in the US. And I also want to mention here that with respect to COVID, because we have this emergency situation, the EMA was very flexible and provides now also a kind of rolling review. Additional incentives, okay, for ATMPs, you heard we have classification, certification for module 3 and 4, so non-clinical CMC information, the hospital exemption. So if you want, we can discuss this in the Q&A session. However, yesterday was a very nice a panel on hospital exemption in Biotech Atelier already that provided some insights on Bulgaria and Lithuania and some European aspects. So I'm happy to talk about it. So then for other innovative medicines, except from what I said already, we also have the joint advices with health technology agencies. And I think that Annabelle later on is also going to touch these topics. And in some cases, for example, for UK, we have specific national programs like the Early Access Medicine Scheme. So I want just to touch on PRIME. So there's the aim was also to support the development of medicines and unmet medical need and major public health interest, to have an early dialogue with the rapporteur. And for the timing of the request, I would just like to show you this time scale. So SME and academia might have the possibility to apply a little bit earlier when they have very compelling non-clinical data and first clinical data. And then all other sponsors would apply at proof of concept stage or a bit later. So we see in the statistics that there's quite a lot of sponsors. I think it's about 50% which are already in phase 3, and I don't know, 25, 30 in phase 1 and 2. Okay, there are numbers. You can look for all the statistics on the email webpage. What I want to point out is that we almost half of the designations are ATMPs. All of the ATMPs in the PRIME program so far, at least as I found it, are gene therapies, so no tissue engineered, no somatic cell therapy product. And the main indications are oncology and hematology, and some others are listed here. You can see also for the statistics since PRIME was since PRIME started in 2016. So with respect to the special considerations from the FDA, all the programs are addressing serious life-threatening diseases. And I want immediately to jump to the RMAT designation. So this is a program especially for cell therapies, therapeutic tissue engineered products, human cell and tissue products, or any combinations of these. Some gene therapies and xenogenic cell products can also meet the definition. And the program is equivalent to fast track and breakthrough. And in the table, you just see a comparison of breakthrough and RMAT. So breakthrough is for any kind of drug, while RMAT is just for regenerative medicine. And for the main difference with regard to what evidence you should provide. So in the breakthrough designation, you already have to show some substantial improvement based on a clinical significant endpoint over available therapies. While for RMAT, you have to address an unmet medical need in a serious disease or condition. There's a nice page you can visit, Bioinformant, which has all the statistics on the RMAT designation. So you can see how many we have, 24 requests, 3 granted this year. And the nice thing about looking at what kind of products we have here is that we see We see also gene therapies, but we also see, for example, acellular blood vessel products, mononuclear cells, allogeneic dendritic cells. Okay, and here's a summary on the approved products in the US and the EU. And what I mainly want to show is that we have some more products developed only EU, especially the gene products. Most of them are in EU and US, so that is the blue color. And you see also mainly approaching gene therapies in the last years. So this is a list. We don't have to go through this now, of course. It will be maybe interesting when you look at the slides later. Just what I really want to concentrate on, this is now only the gene therapies. And you see when looking at the incentives, orphan drug medicinal products, use of prime, breakthrough, rare pediatric disease designation. And when you look at the next slide for companies on the cell-based area, you see 3 orphan designations. You see no other incentives. At least you see 2 conditional marketing approvals. So that was for Holoclar and Salmoxis. So it's very, very obvious that there's there's quite some difference in strategy. So here's also a summary of orphan gene somatic tissue engineered products, also showing we have much more gene therapies than others, quite a lot of orphans. And I just want to mention that I didn't forget that USA has also 8 cord bloods on their list of approved cell and gene therapies. But as I said, that wouldn't have cover the European definition, so I took this out. 5 products unfortunately left the market, and that was mainly for commercial reasons and partly also reimbursement issues. In case of Provenge, there was also a competitor coming about 1 year later on the market, small molecule, so that was a very difficult, of course, then competing situation. Ongoing at the EMA, we have 7 or maybe even 8 candidates now. Again, almost gene therapies. We have 1 tissue engineered product, and we see that most of them also submitted to the FDA, usually a little bit earlier than for EU. And maybe also we like to discuss a little bit about our gene therapy. So this is just some point that I mentioned that I think would be interesting to mention. So of course we achieve very high prices with gene therapies, usually more than 500,000 euro now with what was it, Zolgensma, we have $2 million. We have, of course, with gene therapies very often, at least when it's targeting monogenic diseases, a cure instead of a treatment. So that would be the case for the treatment here mentioned in the brackets: beta thalassemia, ADAS-KIT, spinal muscular atrophy, and the others mentioned here. We also have the cancer treatments, which cancer is the most, the widest indication that most research is going on here. I think from a manufacturing point of view, and I think we'll hear about this certainly later, gene therapy might be a bit easier in some cases to characterize, and there's clear mode of action, and with respect to potency, that is of course also important, especially with tissue engineered products. That is very difficult from time to time. So we will see what would happen to tissue engineered and somatic cell therapies. So of course, those targeting also rare diseases have more possibilities to use also the incentives. On the other hand, especially, well, these might also be able to address broader populations. I mean, we have all the cartilage defect products. There are quite some products, at least in the hospital exemptions, for skin burns, back pain, osteoarthritis. Maybe I can mention here that we have a big Horizon 2020 project called IP Spine, investigating the use of induced stem cells and then notochordal cells for spine disc regeneration. Yeah. So, and just here mentioning, of course, their new developments, and the ATMPs are new, of course, but there's still ongoing development always. So we have the gene editing methods that are included in the clinical trials now. I mean, no drug is authorized yet. There are mRNA products and nanoparticle-coated gene therapies. And we also have products which are not ATMPs, but kind of interesting and from a point of view that some companies now more concentrate on exosomes from mesenchymal stem cells, for example, and trying to get the same effect they would get with the mesenchymal stem cells, but maybe a little bit easier. And also on the base of decellularized tissues that can be personalized. So then I also come to the summary. So we've seen 5 of 15 authorized products left the market due to commercial issues. That was the case in Europe. In US, all of them are still on the market. We have similar marketing approvals in the EU and US. And we see mostly gene therapies and therapies in orphan diseases approaching the market. More or less parallel development, US and EU, with US usually coming a bit earlier. Then mainly gene therapies use the incentives we have. And because I think I forgot to mention this before, I mentioned this here, that for in Japan, so the PMDA there also has a similar incentive called Sakigake, which also provides a very early support to companies developing regenerative therapies. Okay, and tissue engineered are mainly non-orphans, have no incentive, are using no incentives usually. And so I think I'm 2 minutes late, but almost. So thank you very much, and I would be happy to have questions.
Zaklina, thank you very much for the great time management, actually, to you and to Violeta. Thank you for that. I don't know if everybody, the participants, is keeping an eye on the chat box, but if you're not, I want to tell you that Mrs. Maydell had to leave the event because of some engagements she has taken already. So I want to thank her once again for the participation. And in case you have any questions that are addressed specifically to her, or recommendations or comments, we will share with those with her later. After those 2 presentations, I want to tell you that we have foreseen a 15-minute Q&A session, and after this Q&A session, we will actually have a 30-minute lunch break. And now, I don't see any questions from the audience, but I would like to ask a couple of questions each one of you, Violeta and Zaklina. I will start with Violeta. Violeta, you showed in your presentation that actually the ATMP development in Europe is not so enhanced as in other regions. Do you have any comments on that, or have identified for yourself any particular reasons that you think have any relation with that?
That was one of the main concerns. I was hoping it would stay with you, and I believe Buljovcic.
Violeta, we don't hear you.
Oh, Can you hear me now?
It's better, yes.
Oh, sorry. Yes. Good. So I was just trying to say that both Zaklina and I, we have already alluded to some of the reasons this could be the case. But just from my own experience, the most frequent reason which I have seen being cited in surveys with ATMP developers for not having the same level clinical development in the EU with ATMPs is that there is a lot of clinical trial complexity here in Europe, and it's largely due to the various approaches of various medicines agencies included in the various stages of the procedure, although we do have a centralized procedure. And although, for example, if you look at scientific advice specifically, even though it is possible to obtain centralized scientific advice, usually companies have on average to wait for that about 4 months. And the timelines in the US, for example, are only about 60 days, so it's half. I see Zaklina want to comment. Do you want to comment now, or are you coming later?
You can finish and I can comment then.
Yes. So I wanted to also point out that specifically for SMEs, this is a huge complication. And it's obviously delaying clinical trials for sure. What we want to see and what we hope to see is that the scientific advice is rapid and truly centralized, optimized to a greater extent. Another factor, I think, which is really playing big in this field, and you mentioned it too, Miglena, are the GMO requirements for clinical trials, specifically with gene therapies containing or consisting of GMOs. Again, we have various regulatory procedures. Companies have to go to different agencies in the various member states which interpret the law in a very different ways. And again, over there as EuropaBio, we're really at the point of exploring with the European Commission whether there could be some possibilities for streamlining that process and rethinking really whether the GMO requirements that we have applicable in the continent are truly made for medicines, and specifically for this very innovative type of medicines. Another thing that I could mention is probably, and I believe there was also a session on this specifically in the Biotech Atelier, the use of real-world evidence. So that has to be advanced as well. And I think also here Europe has actually a wonderful opportunity because there is no guidance, there hasn't been guidance yet on the use of real-world evidence. So we can be a pioneer in this field, and I also truly hope that this happens. On financing, you've seen the situation is looks good, but again, Europe is not comparable to the US where the money is. And in terms of total financing, the US is still number one. And I know a lot of effort is put into this, and we're in very close contact also as an association with the venture capitalists across the continent. But again, there is a lot more to achieve to really keep pace with others. Yes, and I'll probably stop here because I think it's a multifaceted question and the discussion.
Thank you very much, Violeta. Zaklina, please feel free to comment.
Yeah, so I can maybe add some things to this. So the first one was you were mentioning scientific advices and the delay in time. So I could say, for example, if we look at the German authority for ATMPs, the Paul-Ehrlich Institute, it depends on who's responsible for which kind of products. So it usually is the case that for tissue engineered products and somatic cell therapy products, you don't have to wait more than usually, so that you can get dates within, I don't know, 1-2 months, maybe, rather 2 months. But for gene therapies, of course, there are also people which are now working with all the COVID-19. So it's the same personnel. And of course, there's some delay then due to COVID-19 scientific advices, which are, of course, have a higher priority at the moment. With regard to GMO, I mean, I absolutely agree with you. That is still a problem for the investigators to align their GMO and well, other guidelines, and there are somehow I think it's already a little bit improved also with respect to the agencies that need to be addressed. So I know, again, for Germany, for example, that we have two agencies. One agency, the BVL, is for the GMO, is responsible for the GMOs, and the PharmaLex Institute for the clinical trial applications. But they centered it so you only have to communicate with PharmaLex Institute, and they do the rest. So that's already a little bit easier than before. Yeah. So on Miglena, just that is something I wanted to add to what Violeta said, and I just have I have one interesting information. Maybe if we have the time and it's okay for you, but I would just
Yes, we do.
To take this as an example, because it's really very, from a strategic point of view, very interesting. So if we take Zolgensma, the latest gene therapy that was approved in Europe, so for the treatment of spinal muscular atrophy. So Zolgensma is for small children under 2 years, and as most of you probably know, there was or is another product already on the market, Spinraza, which is also for SME, which is an oligonucleotide. So not a gene therapy, but from the mode of action, you could say somehow comparable. So, however, Spinraza has to be given several times. So every, I don't know, 3-4 months or something like that, and Zolgensma should be given once. And then we have the discussion about the costs, right? So the cost of Zolgensma 2 million, but the cost of Spinraza is I'm not quite sure. I think 100,000 or something around that for one injection. So so that is of course something that has to be considered, and certainly has been considered now during the also HTA discussions. I'm sure we will hear about that again also more later from Annabelle, and there's again, I think, also a third product approaching, and even Spinraza, the companies, so Biogen and Novartis are trying to, as far as I understand, to work together, like that Spinraza could support or Zolgensma. Well, anyway, so what I just want to say is it's so important to see the competitor landscape as well, and that is what we've seen with Provenge, what was one of the reasons why they had to leave the market. So this is a positive example now, looking at it from the view of the gene therapy. So.
And if I can come in again very briefly on price, because I certainly very much agree with Zaklina, and just wanted to say that again we have to rethink here to adapt to the ATMPs, because as we've been organized so far, we've been paying for sick care. Our healthcare systems are organized to care for sick people, and we have to start really paying for healthcare. And again, as I've mentioned, we have now a wonderful opportunity with these therapies to cure patients for life once. So this potential has to be realized, and this is why all the things that we've mentioned before with Zaklina has to be paid enough attention to. And I'm very happy you gave the example with Germany, because we also consider this as a very well-functioning country in this field, and specifically on the GMOs. We hope many more countries will follow the example of centralizing the procedure, and so that companies go to one authority.
Thank you very much, Violeta and Zaklina, for these comments. As we have 2 more minutes, I would like to add another comment from my side and ask you if you can just comment with very briefly. In your presentations, Violeta showed that currently practically USA is responsible for half of the ATMPs that are under development. And Zaklina pointed out that currently Europe has granted 15 approvals in comparison with 10 approvals in the US. So do you think that this will practically change soon, or because of your regulations, practically it can be considered a little bit more efficient, if I can say so, if it's not too bold?
So maybe may I start, Violeta, and you?
Yeah, sure.
Okay, so then I have maximum 1 minute for my answer. So yeah, so I mean, as I said, it's 18 in the US, right? But it's just that the cord bloods are excluded because we wouldn't consider them ATMPs. So I think really that this gene therapy environment where a lot of money and a lot of investment has to go is a good one in the US, and a lot comes from there. So and I think it will be going on like this so that we have a parallel development, more or less. But you started, I think, kind of a little bit earlier, so therefore I think we have this first additional approvals. And as I say, I hope that also some tissue engineered and somatic cell products will come in between. Violeta.
I also believe both geographies will be moving at more or less the same pace when it comes to securing patient access. So I wouldn't expect very huge divergence really in the number of therapies which are going to be approved, and also because they're actually cooperating very well and strongly with each other. But what is different, as you also alluded to, Zaklina, is that what is important to see where these therapies are being developed. And we see that most of them are being developed in the US, because this is again where the access to finance is much easier, and that's a very, very important condition, especially for very small biotech companies. And you know how the biotech model is, a business model is actually functioning. They have to be run for years without making profits. So having this capital and investment very strongly represented throughout all these years is very important, and unfortunately we cannot match that in Europe yet. And then in terms of the regulatory processes, in which will be more efficient, again, I think the regulatory cooperation between the two authorities will rather move us equally, I hope. And you've seen also the announcement that in Europe we can now expect to have the so-called BARDA agency to finance and advance research. So we remain very hopeful and optimistic.
Thank you very much for answering those questions, but my colleague Paco, who is supporting me with the technical part today, has also a question, so I will take the liberty to give him a few minutes to ask his question now. Paco, please.
Hello. I don't want to interrupt you. That's why very interesting questions, and thank you very much for the presentation. First of all, excellent one. I have just one question, which is actually related to what Zaklina mentioned regarding the Zolgensma and price, which is one of the big problems. And my question is not only to Zaklina, but to Violeta as well, because Violeta also partially mentioned this in her presentation. So the cost is from one side probably one of the main reasons why the HTA and then the reimbursement is so slow and so hard to be achieved. Do you think that the price is reasonable? And don't you think that actually, how to say, is more or less manipulated the price due to the fact that this is a unique product, no alternatives, and the patients, they don't have actually alternatives yet? Yeah, this is my question. Thank you.
Okay, so you asked me now, because I'm sure that Violeta also wants to say something. Okay, so I mean, yeah,
it's not a straight question. I just want to, yeah.
Yeah, I mean, it's. I mean, I'm not to judge the prices of the product. So what I mean, the price of the product always has several components. So one component is the manufacturing, all the costs that the company really has, and the employees, etc., transport, and so on. And of course, there's also costs on the disease, so that an orphan medicinal product can can ask for higher cost also to get all their investment refunded. So and I think partly we also come to an ethical discussion on, well, where the price then really can be, or if a company maybe can ask for less, or if it's already very low but we think it's very high because it's an amazing number. Yeah, so that's all I would like to say at this point.
Thank you. Yeah, the balance is the key.
Maybe very briefly also from my side, Panko. So what is important here is really to capture the value of the therapy to that individual patient. And this is very, very difficult actually also for people good with numbers because the value for individual patients is different. For every patient, it's different. And then I don't know now, people are working on methodology for how to assess those, and I don't have the numbers right now at the back of my mind, but I can share a study which has been done on a therapy against hemophilia, because companies do realize that they have to show some statistics here, how you can actually because this is the very high upfront cost, but then it actually distributes then further on in years. Because as I said, we are now paying for sick care because these SMA patients, if they survive for long enough, they will be cared and money will be spent, unfortunately, on them for years and years. And actually, there are alternatives to Zolgensma, as also Zaklina mentioned Spinraza, and the company here in this case, this is how they also came up with this price, because they took the 10-year base for when you have to care for an SMA patient for the period of 10 years. So it was not just a number which came out of the blue, but again, a lot of work has to be done here, because indeed we also appreciate from our point of view as an association the importance of explaining to people why the prices are, why the upfront price is so high. But we believe there are many advantages further down for the entire sustainability of the healthcare systems altogether, especially if we can provide with ATMPs many answers to many of the chronic diseases today, like for example cancer and the various forms of cancer.
Thank you for the answers.
Thank you very much, Franco, for your question. Thank you, Zaklina and Violeta, for presentations, for answering the questions. And welcome back after the lunch break. Now we will continue with the presentation of my colleague from PharmaLex UK, Adeyemi Afuwape, who is Associate Director of Regulatory Affairs, CMC. He will present to us the non-clinical challenges and cases with ATMPs. Georgieva, are you online? Do you hear us?
Yes, I am. Thank you very much, Miglena. Yes.
Please, the stage is yours.
Okay, thank you very much. Thank you to all the organisers for basically inviting me to this meeting. Hopefully we should be able to cover a lot of great things. So I'll be looking at the non-clinical challenges for ATMP. Okay. So yes. So the first thing we're going to talk about is the kind of development of preclinical or non-clinical development as far as ATMPs. So non-clinical and preclinical covers a wide range of studies, which covers either in vitro or ex vivo experiments looking at toxicity and pharmacology, or they can be taken the guise of computational or in silico kind of simulations of interactions. Now, preclinical and non-clinical can be used, terms can be interchanged quite frequently, but preclinical studies really essentially cover the studies that are used before any clinical trials and to support the evidence for clinical trials, whereas non-clinical development looks at studies which covers studies which does not involve a human subject and can basically cover across the whole product development line. So preclinical, especially if you use animals, most of the principles is on the regulatory principles is on the acceptance of what's called the 3R principle, and what it encompasses the ethical use of animals in studies, and it covers a way of looking at ways to replacement, reduction, or refinement of the testing product. Now, non-clinical development, as I said, really can be divided into two: exploratory studies and regulatory studies. The exploratory studies obviously are the ones which are used for kind of justification of clinical trials in humans, whereas the regulatory studies could basically span the lifespan of the products, and can be interjected while the clinical studies are in place, and even beyond market authorization. So, clinical development, why is it done? Well, it's to provide an understanding of the drug's mechanism of action, provide evidence that the drug is reasonably safe to conduct the proposed clinical trial or investigation, inform the design of early stage clinical trials. That's looking at things like the starting dose, dose escalation, dosing regimen, and route administration. Guide the patient's eligibility criteria and safety monitoring procedures. So it actually effectively kind of provides reasonings for that, and identifies risks that aren't readily identified in human trials, carcinogenicity and reproductive toxicity. But the main purpose is to understand and minimize the risk to which the humans are exposed. And what you really want to do is a good non-clinical study will actually identify the risk and identify the risk and toxicities that may happen. If you don't identify that, then maybe the design of your non-clinical or preclinical studies are not necessarily answering the questions that you require to move forward into clinical trials. So non-clinical development and regulatory framework, especially in the EU, basically is kind of underpinned by the ICH guidelines for non-clinical safety studies. And it is true for the EU, US, Japan, and other regions. Non-clinical development then also has a regional way of regulating these trials. So obviously with the EU, it would be the EMA. And fundamentally, across the regions, when you go for your non-clinical development and preclinical development, but non-clinical development, especially when it comes to toxicity and safety, they have to go through what's called GLP, good laboratory practice, and this is a legal requirement for you to undertake these studies, doing set of principles, and it's essentially a quality management system rather than a scientific system, but it's essential and a legal requirement for any studies that are going to use to support the justification in human trials. So, a non-clinical program study design is a general overview. Non-clinical studies should resemble the clinical setting. That means the administration should be the same, the frequency of the administration, and the duration of the clinical study and the treatment. So there has to be a way of that the clinical program that you have has to resemble what you intend to do in the clinics. So there's general considerations for small molecules. There is 2 species, 1 rodent and 1 non-rodent, which cover both male and female. This is also true for biotech products, 2 species for short-term studies up to 4 weeks and 1 for long-term studies, male and female. It's considered that non-rodent animal models are more informative than rodents, but if there's no relevant species for your study, homologous molecules or transgenic models can be considered, and this is pretty important, especially for ATMPs. As I said, the safety and toxicity studies support clinical trials should be conducted according to GLP, and there is not a requirement for the material that is used to be GMP material. However, it's expected that the material that you are using is representative of what you consider to be used or what you tend to use in the clinic. This is particularly important for biotech products and those that look at novel formulations. So, with a non-clinical program, it's subdivided into pharmacological studies. The primary pharmacology is looking at basically the mode of action, the effects on the related desired therapeutic effects. The secondary pharmacology looks at the effects that are beyond what you consider to be the mode of action. And then the safety pharmacology what's called the core battery, and that looks at the effects of systems like the central nervous system, cardiovascular, and GI. Then you have the toxicity studies, which are really looking at the safety aspect, and they're most exploratory and very important for any studies to support first time in human trials or pre-IND or IND enabling trials if you're looking in the States. What's also important is that the ADME studies look at exposure, absorption, distribution, metabolism, and elimination. And these are very important pharmacokinetic indices that has to be looked at in all studies. However, if we were to look at ATMPs, these are not necessarily, they're not applicable in most cases. So absorption, metabolism, and elimination cannot be directly applicable to a lot of ATMPs or cell gene therapies. That's because, for example, for CAR T therapies, the T cells have the ability to expand in vivo, to respond to tumor changes, to respond to tumor or antigen load. So if you induce or you administer these cells will basically go, they will deal with whatever antigen specific response to a tumor load, and then they will quiesce, and then basically the response will recede. However, if there's an increase or another introduction of the antigen or tumor into antigen, these cells will basically expand. It's considered to be a living medicinal product. So absorption, metabolism, and elimination is not going to be appropriate. Likewise for associated adenovirus therapies, where your transgene is basically long-term exposure, especially in local areas. Again, it's not the elimination or removal of the transgene and the virus is not necessarily going to be follow the classical aspects. What is important in both of these cases, and especially for gene therapies, is that you've got to measure the level of persistence. Persistence is going to be an important factor to show what the safety aspects of your therapy and what the impact it may have, and that's going to be a very important indices for you to measure and to present. Fundamentally, the most important thing is the optimized development strategy to underpin a clinical development plan. And the important aspects is the timing of the non-clinical study in relation to the clinical study. So really, what you want to do is make sure that the time that you plan your clinical study, whether you've got enough information and material to be able to justify and present to a PIND or a scientific advice meeting of your clinical study. The choice of the animal species is vitally important, and the selection of the doses in toxicological studies. And required non-clinical studies, some recommendations and rationales for study and design and relevance of the clinical studies is very important. And so therefore, an early interaction with the regulators is going to be pretty important, particularly for cell and gene therapy products. And lastly, with the role and timing of regulatory agency inputs. And that means very early interactions or expedited programs is at your disposal, whether you go through the EU or the FDA. So, in a classical non-clinical study or preclinical study, what you really want to do is find what's called the minimum acceptable biological effect level. And this is justified by the pharmacology, adjusted for the exposure in man. And what this is basically the lowest part of the therapeutic range of your drug product. This is really important for justifying the starting dose in your first time in human. You have to then obviously make sure you look at the toxicological limits, and the toxicological limits are basically the no observable adverse effect level, NOAEL. And this is basically the lowest point where you would expect to have unacceptable toxicity. And in that, between that window, between NOAEL and the MABEL, you are then looking at your starting dose, the recommended maximum starting dose. When you take into consideration the toxicology has to be adjusted for the anticipated exposure, and obviously whatever species you use, there's going to be an adjustment and a safety factor. However, again, this is not necessarily the case for ATMPs, especially for CAR Ts or AAVs, but especially for CAR Ts, because again, as a living medicine, the dose that it can be used could be really dependent on the response of the cells to the tumor load, the clearance, the immunological environment that the T cells are presented. And even particularly more important for an autologous situation where you are using very personalized T cells. And that means that the dose variability will be dependent on not only the manufacturing of your cells, but also the actual immunological status of the patient. So the clear case of minimum anticipated biological effect level is not going to be totally applicable to the T cells, and so therefore the design of your non-clinical studies have to reflect that. So there are a number of non-clinical guidelines for ATMPs in the EU, and these are quite a few. So there's three of many which actually looks at the aspects of gene therapy medicinal product guidelines, which covers the quality, non-clinical and clinical aspects of gene therapy products. There's obviously a reflection paper on the design modification of gene therapy products. This is particularly going to be important and relevant for early stage studies, and the guidelines on non-clinical studies for first clinical use of gene therapies. And that's going to be really important to underpin what justification and what is required to start your first time in human trials. There's another set of guidelines which looks at the things, the risks. So you have guidelines on the scientific requirements for environmental risk assessments for gene therapy products. And this is really important to link for called GMO assessments, which is going to be important for you when you submit your clinical trial. And guidelines on non-clinical testing for inadvertent germline transmission. And this is basically for gene therapy products, the risks or the assessment of the risks of gene therapy or gene interactions with reproductive organs and cells, gametes, because obviously that's the safety aspect of passing a treatment through to to children and progeny is very, very, very important, and it's a very important safety aspect of any gene therapy trial. And then we have two specific guidelines and reflection papers. One, which is basically looking at the aspects that we needed specifically for adenovirus, AAV vectors, principally used in vivo gene therapy. And another one is basically looking at the medicinal products containing genetically modified cells, where these are the ex vivo genetically modified, and this is your CAR T cells, your NK cells, and TCR modified T cells. So if we look at the safety concerns that surround gene therapies, the first thing that we look at is mostly the type of vector used for transduction or the virus involved. And a majority of the vectors that are used in gene therapy until recently is being basically lentivirus and retroviruses. And these are integrating vectors which introduces the gene of interest, your transgene, into the genome for essentially more permanent kind of expression. These have very important safety aspects. You have to consider basically whether they will cause any kind of mutagenesis or carcinogenesis, whether they will integrate the gene close to oncogenic sites or known oncogenic sites, and also whether they will destabilize the genetic stability of the cells themselves. Whereas with adenoviruses, they are not considered to be, they are non-integrating, so they don't integrate into the genome. They are, on the whole, not considered to be pathogenic to humans, but and they are transient. But the most important thing is really that the tropisms, they're different serotypes of AAVs, and they have different affinities for transducing tissue types, and there's a wide range of different type of AAV. So you have to look at the different AAV serotypes that you would need for your particular therapy. Vector virus distribution to non-target cells is very, very important. And that could be governed by the route of administration, whether it's local or systemic. Principally, if you look at AAV, you really got to look at how you can actually measure that the administration of your in vivo therapy is contained to where you expect it to go. and if you're doing it systemically, how you can monitor that and what safety aspects is required. It's important to look at the persistence of your transgene as well as the vector if you're using. So therefore, there's two levels of way we look at persistence. You have to have a guide and an understanding of where your transgene is being expressed and the vector itself. On tumor, off tumor, on target, off tumor tissue effects. That means that specifically for CAR T cells, where your tumor antigen is on your tumor, but there's some level of cross-reactivity where this antigen has some level of cross-reactivity on an expression, low-level expression on healthy cells. That's really important. So you have to make sure that you do an assessment on the relative binding of your short chain variable fragment that you use for your CAR to make sure that the CAR that you subsequently use does not have any cross-reactivity on healthy tissue. As I spoke to earlier, germline transmission, again, with gene therapy, anything with gene therapy is that you're trying to inhibit any response in reproductive organs and gamete cells. And also, as I talked earlier, the species-specific AAV serotypes, because again, in some regards, if you're looking at that, there is you may have problems using a particular AAV serotype in your model, which is not directly linked to the human situation, and you may have to change that to a more appropriate AAV serotype in your model if you're using for mouse and the tissue type as well. And that's going to be important for safety concerns because you need to have you need to make sure you can model what you would expect to see with the human form or the human the human situation with the AAV you're interested in. Level of replication, bioreplication, and persistence is vitally important. So when you're using lentivirus and retroviral viral vectors, you've got to look at the capacity or the potential of them to form replication competent retrovirus or lentivirus. And you've got to look at that potential. So therefore, even though when you're designing and you're making your virus, it's spread over a number of plasmids to generate your virus, and you've got the safety aspects, for example, self inactivation, LTR, long LTRs. You still have to check that there's going to be a possibility, however low, that you could actually generate replication competent lentivirus or retrovirus. Another aspect is looking at viral copy number. You have to measure the viral copy number in your cells to see when you transduce them, what level of viral copy number is transduced in your cells, and you have to have a measure of that because that can actually implicate not only the safety of the product, but how it moves, the potency, and also any kind of toxicity that you generate. Inappropriate immune activation by the vector and transgene is pretty important. If you have an AAV or that is tissue specific, you want to make sure that this transgene activity is in a specific area, and if it obviously moves to a different part of the body, what level of immunogenicity can that introduce? The transgene itself, what you're producing, how immunogenic is that? Whether it's basically the immunogenicity that's induced by this transgene will offset its actual pharmacological or therapeutic effect that you look at. AAV serotypes is really important because there is a large proportion of the population who have antibodies against AAV, so the serotype that you use could actually be important, number one, for how you introduce which patients that you'll be able to include in your trial, but also how it is effectively the efficacy that could be done. And if you do repeat administration, that could actually affect the efficacy as well as the safety aspects. And finally, third and fourth generation CAR Ts. Now, third and fourth generation CAR Ts are CAR Ts which have got the CAR receptors externally, and internally they've got all the different level of transducible domains, which does activation and persistence. But in turn, they've now lots of cell therapy developers or gene therapy developers are introducing other aspects like cytokines, like membrane-bound IL-15 or IL-12, or basically knocking out checkpoint inhibitors. What this means is that the T cells that they're using not only has their antigen-specific response to the tissue-associated antigen, but also are modifying the microenvironment by expressing cytokines or stopping inhibitors. And you have to it's really important for you to check and assess that in your model to make sure that these are not introducing any level of inappropriate or unwanted or undesirable immunoactivation over and above the antigen-specific response you're intending to use. Another potential for kind of safety aspects that should need to be considered is vector insertion or mutagenesis. And as I said, retrovirus and lentivirus are integrating viruses, vectors, and they introduce the gene into the genome. You have to check to see where the gene is being introduced or integrated in your genome is not anywhere near any oncogenic sites. And you can do that by either computerized and in silico assessment, so looking at the publications and seeing where these oncogenic sites are and seeing how the publicated data about that, or you can do some whole genome sequencing, where effectively you are looking at to check the oncogenic sites, comparing that with transduced or non-transduced cells. There's been an increase in induced pluripotent cells being used for generating not only new tissue, but also immune cells, CAR Ts and what have you. And there is a real safety concern about the tumorigenicity that these cells can induce. And you check that with in vivo murine models or in vitro systems looking using matrigel, looking at plaque forming and tubular forming structures. And these in vivo models can take up to 9 months to look at the assessment of these cells' potential to actually form tumors. With genetically modified cells, you look at for inappropriate cell proliferation, differentiations, migration. Now, I've put in here T cells and NK cells because NK cells are end-stage cells. They are less likely, they're not long-lived in vivo, and so their safety aspects does not compare in this instance. Whereas T cells, they have the propensity, as I've said before, to basically self-replicate and proliferate a number of times. And these are because these have stem cell-like phenotypes. So you've got naive T cells, central memory T cells, or stem cell like central memory T cells. And that means these cells have the propensity and the ability to replicate and basically replicate in vivo for a long period of time. And so you would definitely need to look at biomarkers for systemic cytokine release in that regard. And that's going to be an important factor in your non-clinical studies and your models. We have residual non-differentiated iPSCs, which if they are used to generate these T cells, or even tissue cells, you want to make sure you eradicate any undifferentiated iPSCs, because they themselves, if you have them in your product, they would effectively induce tumorigenicity and what have you. What you would want to do is to basically look at a model where you look at the lowest number of, or basically the highest number of cells which will start inducing a tumor effect. So you've got to look at the minimum maximum levels of these cells in vivo. TCRs and CAR T cells, because of their propensity to cause cytokine storm, there has to be control. Several developers are introducing what's called suicide switches on these cells for them to actually be able to control and eliminate these cells if in vivo there is a toxicological event. They either use a small molecule like a remidusib, or they use a monoclonal antibody, licensed one like rituximab, where they introduce the receptors for those agents on the T cells. So if they, in the likelihood that there's a toxicological event, they will introduce that to remove the cells. We have also viral shedding, which is really important. That has to be assessed and shown, because that's going to be an important aspect of especially for in vivo gene therapy. You've got to look at your devices, whether it's retinal, cardiac, and intracerebral. You want to look at maybe the risk of viral shedding in that regard. And this data actually goes forward towards your GMO assessment in the EU, or the LMO, or living modified organism in Japan. So these safety aspects on your non-clinical data actually will contribute to your GMO assessment. A specific gene editing technology has been on the rise with CRISPR, zinc finger, and TALENs. These are technologies which introduce a double strand break in your DNA to introduce either a cutout, excise, or introduce a gene of your interest. On-off target effects is really important, so you can check that these editing technologies are cutting at the right specific place where you want it. You also have to look at translocations. Once the cut has been made and there's basically gene rearrangement, you've got to make sure that the gene rearrangement is not a safety aspect. You can check that with fluorescent in situ hybridization, and you can use karyotyping for genetic stability. The final thing would be the immunogenicity assessment for nucleases is really important, especially for CRISPR, because it's bacterial derived, and so therefore immunogenicity could be a factor. So let me move quickly on to the case studies. So,
Yemi, I'm sorry to interrupt you, but with regards to time management, I want to remind you that we have probably up to 3-4 minutes for the rest of the slides. Thank you very much.
I will go straight on to the last two slides. So, in 5 of the CAR T therapies, pharmacodynamics, as I said, there's a lack of models that can generate relevant PD data. You have to really look for that. So you have to choose your model carefully. Once at a time, you've got to know the identification of what clinical setting you want to put in your model will show, and rely on surrogates. Obviously, pharmacokinetics has to be dependent purely on biodistribution and persistence, and the toxicity and safety has to be on vector integration related effects for your vectors, off targets, check the off targets, and also immunogenicity. So the final slide is basically on the non-clinical development of 2 known CAR T therapies, QnA and Yescarta. In both instances, what they did, they looked at specificity of the CD19. QnA, they used basically in vitro, they used the CD19 CARs and looked at the binding efficiencies on membrane protein assay, or could be surface plasma resonance, and they're looking at the tissue binding affinities for that. While the Yescarta, what they did is they used a co-culture system with anti-CD19 CAR T cells from patients and looked at that in an in vitro system to look at the efficacy and safety of basically the mechanism action. For distribution, QnA, they use an anti-tumor activity in xenographic model. So basically, immunocompromised mice, they introduce the tumor and then introduce the cells of interest and see whether they can actually have the killing activity and how they're distributed in the models. Yescarta, what they did is that they used an analogous model. What they did, instead of using human T cells, they used murine T cells with the CAR for targeting CD19 or murine CD19 in their mouse model to replicate what they would consider to happen in the human situation. And finally, for pharmacokinetic and toxicology studies, QnA, what they did, Novartis used, is they looked at tox parameters, cell distribution, persistence in the models, again, looking to see whether the safety aspects of that. But also they did a genome insertion site analysis of the lentivirus that they use in the genome. So what they did is that they used the lentivirus, they compared transduced and non-transduced cells, and then did the whole genome sequencing to check that where the transgene was being inserted was in a safe place. Kites and Gilead with Yescarta, they did that, but they did it on review publishing data and clinical information, and they based that on the information that was used for T cells that were done with the retroviral vectors, which they used in their product. And sorry about that, and that's the end of my talk, and hopefully it wasn't too long. Thank you.
Managing the development and life cycle of a pharmaceutical product can be beyond time-consuming, beyond costly, and beyond complex. PharmaLex enlightens with trusted expertise and empowers with cutting-edge technology. PharmaLex ensures healthier business decisions with specialized services that are adapted to fit your needs. Scalable yet approachable and innovative. Our services extend beyond market approval to include product maintenance and post-launch activities, both at a local and global level. PharmaLex, beyond knowledgeable, beyond flexible, beyond innovative, beyond trusted. And with over 90% of business from satisfied repeat customers, beyond proven. Let's take your business above and beyond. Contact us today.
So thank you for having me. My name is Zeb Younes. I'm a Director of Reg Affairs CMC. Thank you for the introduction. And I will be talking about ATMPs, tissue, cell, and gene therapies in terms of CMC challenges and case studies. And before I really get into this, I will also go into the drug development pathway, but from a CMC perspective. So the topics that we cover fall into place. Before I really get into it, let me try and clear my screen a bit. Here we go. Before I get into it, I do want to clarify that the opinions and information in this presentation are my own and do not necessarily reflect the views and policies of the company. The information here consists of examples, and they are examples from my own experience. And I want everyone to be aware that I may give examples, but they are just exactly that. There's always differences to every rule and expectation, as these products are diverse and complex, and they are judged on a case-by-case basis. So I'm going to start with a somewhat detailed discussion on the drug development pathway from a CMC perspective to emphasize some of the key aspects to consider during development. And then I will go into as many case studies as I can, that time allows, and then summarize the main concepts at the end. Now this slide, this is a busy slide, but actually it's one of my favorite slides because it illustrates all the points I want to make in one place. Before we really break it down, let me just talk you through what you're looking at. We have these 3 arrows along the top, and they represent the different stages of drug development, where you have the preclinical activities, we have the clinical development, and the post-approval activities. The size of the arrows does not represent the time these steps take. It is only a reflection of the space I wanted to convey my messages. Along the top, we have some activities that need to occur during development, and along the bottom, we have all the CMC activities that I want to emphasize. In particular, we're basing it around material that's manufactured and the work that needs to be done on this material to get through these phases of development. We have these little gold stars here, and these reflect the different points that agency meetings typically occur or should occur, and they're usually just before or just after clinical studies. Now, I'm sure you've heard the clinical programs for these products can be really varied, and given that many of them are orphans, unmet need, significant benefit, the trials can be reduced scope. So I've just assumed for the purposes of this discussion 2 studies, but there may be, of course, additional layers of complexity. And then we also have a meeting that should occur pre-BLA for the US or pre-MAA for the EU. Before that, there should be a pre-submission meeting to really get into some of the topics that I'll go into shortly. And then after this, in post-approval, of course, CMC doesn't end. So we'll cover this. Now, in addition to these 3 slides, we have a dedicated arrow along the bottom for comparability. And this starts from the point initial material is available, and it works continuously through development and post-approval. And I'll consistently refer back to this. Okay, so I think we can now get started. The other point I should make actually is that although I'm going to cover CMC development activities, it is expected and standard that the quality side of these activities for novel reagents, excipients, intermediates, placebo, any finished product kit components and devices and presentations, all of this also has to be accounted for from the CMC side. So we begin right at the start, ideally, but not usually, to be honest, with a target product profile. And this, of course, is a live document that's updated after each milestone during development. This is important even just from a CMC perspective because it prevents project creep, scope creep, and if your drug development team, your manufacturing and formulation folks can see this, then it helps keep them focused because this should emphasize important information that we as CMC people need to know, like the intended route of administration, and that will help when you're defining formulations, the expected viable shelf life, the acceptable storage conditions, the target quality, just from a top level. It, of course, will become more and more detailed and defined during development. It also includes, of course, the target markets that you're looking into or considering at this stage, which will direct all the CMC folks to the relevant guidelines and regulations in those regions. And it also covers, of course, indications, what you're looking for at this stage, which may change during development. But it's a starting point. It really should be a document that's updated and owned actively during development. The next piece we have here is the risk assessment, another live document updated throughout development. And just as a logistical activity, from my experience, when these activities have sometimes been set up with some real effort and a lot of time, but if they're not actively managed and assigned an owner, they're not maintained. And development for CMC does not change. This is a dynamic landscape. We have tight timelines and material requirements, process requirements, so many activities, so many factors can change, and we've got to stay on top of the risk assessments. So I would really recommend dedicated a person or a team is assigned in each company for management of risk assessments, because there isn't just one. There are various risk assessments, and depending on the design of the drug developer, you may have multiple contractors, multiple manufacturing sites, testing labs, sites that provide novel reagents or excipients. All of this material and information needs to be assessed and stayed on top of. So this is some advice. The CMC manufacturing side, let's see where it begins, where you obtain the starting material. So this includes obtaining starting materials, reagents for cell therapies, particularly donor materials, cell line selection, right at the start. This is where you have a concept and you are trying to define whether this is going to be feasible by generating your first set of material. Even at this stage, you need to consider CMC and longevity of your product. There could be real concerns because at some point when you have some material, you may realize that you used a patented cell line, you used inappropriate material, your material had animal components that can't be excused, it wasn't safe in terms of the virus clearance, and that will mean whatever you do, you'll have to start again. So it's worth doing some basic checks at this stage to make sure that whatever you're going to generate, no matter how early it is, it's controlled and assessed as safe. So you take this material, the starting material, and you begin the process development. This is usually at lab scale under development facilities, various institutions, and this will give you a first understanding of your product. You'll have first time you'll have material in your hands. You'll be able to get an understanding of its quality attributes and how the process operates, the process parameters. This is where you'll first start putting in some formulation development, some consideration to whether you can develop your formulation or whether it will be minimal development and a deep freeze, or whether this is a sustainable formulation for the limited amount of time that you need prior to administration. Perhaps you need to thaw it and hold it in an infusion bag for some time. Then also, to complement this, you'll have learnt your information on the stability studies and hold time studies. After that, this material is actually quite precious, so all of these activities need to happen to this material. You need to set up your methods. So it's very early understanding of the kind of methods you have. A lot of these methods won't be off the shelf. And many CMOs and contract testing labs won't have them just in place. They'll need to optimize, develop them, test if they're feasible and sensitive enough to do the job they need to do. And then you'll also be setting down an understanding of if you're using donors or starting materials, the requirements of these materials. And if they are using donors, you need to understand there are specific requirements. And at this stage, early development stage, you'll be trying to define what those could be and looking into your options depending on which regions you're in. And then, of course, you use this material for proof of concept in vivo studies if that's applicable. And at this point, you've got some understanding now. You have an idea of a process, you have some material, you have feasibility that this could really be a commercial program. So then at this stage, you develop a process that you are happy with and content to use for GMP batch manufacture. And ideally, you'll consider whether the process is scalable. This comes up later, and it's a real issue if not considered early on. Scalable in the fact that here you might be making this in a lab on a bench somewhere, but in the future you want to be able to market this product. So are your materials and components affordable? Are there alternatives in case one of the facilities fails or material suppliers fail? And are all the hold times appropriate and feasible for a running facility at scale? Once you have your material, you generate an engineering batch or could be referred to as a pilot scale. And this is if you're scaling up. It could all, of course, be at one scale. Regardless, you'll generate a batch that's your pre-GMP batch. And this batch will be a test run for your GMP batch to check if all your procedures are in place, if they're working and it's appropriate. You'll use the material from this batch if you're happy that the material from this batch represents your GMP process, which is quite important. If it does represent your future process, then this is the material you can use for a number of the studies that my colleague Adeyemi discussed earlier. For studies like the non-clinical pre-phase 1 studies, the tox studies, you'll of course also use it for characterization. Now you'll really be able to build some data that's going to go into your clinical trial application for the CMC side of characterization of impurities and any stability. You'll have to start making sure that your reagents that you're using are as compliant as possible. Now, there's quite a lot of flexibility we've experienced with the agencies, but we've gone to agencies and we've explained we have a bespoke reagent. There's no alternative out there, and we test it to this level of quality, and this is how we receive it. But the agencies have only ever accepted that when we've been able to demonstrate we've done our utmost to ensure the standard of our reagent is at the best it can be at this stage, and committed to ensure by of MAA or BLA it will be at the standard it needs to be. So you make sure you have compliant reagents as much as possible. And now the analytical methods, you need to ensure they're fit for purpose methods. So this isn't just we have a method, it's we have a method, we've tested it and we've qualified it, or phase 1 validation, or whatever term that the analysis lab uses, but you need to ensure they're fit for purpose. So this is generally quite stringent, except in the case of these kind of products where it comes to those potency methods. There's real flexibility here. And they are expecting state of the art, and it is understood some of these methods may be complex and they may not be the level of qualification you would have for other methods. Perhaps some of your interassay controls, some of the cell lines you're using, perhaps all the controls won't be in place. You need to show it's as fit for purpose as possible at this stage of development, and as a minimum to demonstrate the method does give you a strong indication of potency, so it correlates to the clinical mode of action. And it may well have to be an in vivo method, depending on whether you have any suitable in vitro analyses. And of course, now you'll have to also establish a formal set of donor sourcing if that's applicable to your program requirements. So now you have a process, donor requirements, the right material. You now plug this into a GMP formal batch manufacturing master document, and then you execute and you prepare one of your first clinical batches, which would have to be manufactured to GMP. And once you generate this, one of the first things you do is related to this comparability bar here, is you confirm that you have process consistency, so that your pilot batch or engineering or tox batch is equivalent to your GMP or clinical batch, that the material and the tests that you generated here and all this information you have represents your clinical material. So then that gives you some good leverage to move forward into clinic. You further characterize this material in addition with this new information. This is an ongoing activity. You also put this onto some stability monitoring program, and you make sure you complete either on this batch or in fact a representative tox batch, your in-use handling study. So you have to demonstrate in your clinical trial application, the CMC aspects, which go in the IND for the US or the IMPD for the EU. You have to demonstrate in these documents that you have material whose product quality is unaffected during the clinical bedside preparation. For example, as I mentioned, you may have to take this material, thaw it, reconstitute, dilute, and then administer. And you have to demonstrate that the whole administration process results in no impact to product quality. So this study needs to be captured, and it needs to reflect the whole clinical bedside that you're planning on administering here. And then if one of the other topics that comes up a lot, more often than you would think, is the logistics. As these particular products are so sensitive, and not just the products, the reagents, the materials around them are so labile, the logistics and control of temperature. So you need to ensure that all those shipments are monitored, tracked, checked prior to release for all of those various stages. So now you have submitted this, you have approval of your clinical trial, clinical trials begin and the clinical program goes on. While this goes on, you typically have to generate material to GMP continuously to feed the clinical program. And as this is happening, there's other activities you need to complete in order to be ready for the MAA or the BLA. And this includes building the stability data, so it's an ongoing activity, and modeling it. In some cases, you may be able to model the data that you see and come up with defined controls and trends. Your method development will move on quite far from what it was early to quite optimized methods now. Although they were qualified, you'll now be looking at aspects like robustness of all the variables in these methods to ensure they are fit for purpose as you move forward for commercial. If applicable, you might have to lay down a reference standard for your program, and you might have had to actually do that off your engineering badge. You will, of course, quite importantly for these products, conduct data trending for process consistency. Given, depending on the type of product you have, and they're so varied, it's hard to cover them all, you might not be able to do the traditional process validation, and you might not be able to apply full principles of quality by design. I haven't seen this before, but what you can do is take as many of those as you can and de-risk your program. So if you could implement an early staged, very detailed data trending process, you could actually begin building a detailed model of your process consistency and characterization, which would feed in to ongoing process verification and support the fact that your process is validated without executing the full standard process validation study with all the additional variables. Then we also have, using that information, you can complete your process characterization. And at this stage, if you move that forward throughout, you'll be able to define critical process parameters. And then, of course, you'll have continuing dialogue with these different regions, and they will likely give you different advice. So you'll have to complete different studies for different regions, something to account for in any budget and buffering activities. Then you move into the pre-BLA phase. And at this point now, you need to be ready for commercialization. So we'll have an understanding of what you're going to claim for commercialization. So you'll have characterized critical process parameters, well-established quality attributes, validated, fully validated, including robustness, analytical procedures. Your process will be valid. Your in-use study now will not only be assessed and confirmed, but in use for commercial, realistic commercial preparation by a physician, by a pharmacist. This will be defined and confirmed to be robust. You'll have confirmed formally your commercial release specs, characterization, your impurity clearance, that your process is able to generate material free of adventitious agents, and commercial specification setting based on data and quite tight, or you'll be pushed into tightening these. And then there'll be the region-specific studies that I've mentioned, and of course fully compliant as far as practically possible and agreed with the agencies, starting materials and excipients. One of the key points though, and a lot of companies fall over on this one, and it affects filing dates, is are your facilities inspection ready? So perhaps a facility would be set up quite early on. You may even stay with this facility. You may only realize at this point it's not scalable, or you may only realize at this point it's not inspection ready. Perhaps it has an inspection history, but only for investigational products, which is quite different for commercial products. So this is an important question to ask, and involving compliance activities early on will help you. And then once you have approval, you go into launch. So you may have to do some post-approval commitments to launch a product, but then also an ongoing action that always comes up is variations. So there may be changes to starting, raw materials, test facilities. Some of them may be in your control. Some of them you may have even planned in your MAA or BLA with a upfront change protocol. With others, they may be out of your control. So this is where comparability continues and CMC continues. This is a top-level overview of the drug development pathway. Okay, so I'm going to quickly, I can see I have spent quite a lot of time on that, so I'm going to quickly go through some of the key case studies I have here. Before I get into them, it's important that we understand for all of these, in the case of CMC, we have reduced timelines. Many of these products, as I've mentioned, are orphan, unmet need, significant benefit. And the great news is you may be allowed various benefits, such as breakthrough or accelerated conditional approvals. You may have rolling reviews. The problem is this really crunches it for CMC, because unlike some of the other areas where you'll be allowed to take provide rolling data or generate data post-approval, in this particular case, you can't really skip most of the CMC aspects. Some of them you might be able to provide in a rolling review, but you can't really skip them. You still need to demonstrate your product is safe and that it can be produced to the desired quality. And this unfortunately comes up all the time with many of our drug developers who have great science, great strategies, but the CMC is often neglected because you have to demonstrate quality. Very few quick examples. Miglena, please let me know when I'm out of time. I have.
You still have full 5 minutes ahead, Zach.
Thank you. That's very generous. Okay, so here we have an allergenic cell therapy, and this one had a very complex drug development history that was on and off. It went through various companies, and eventually it was acquired by one of our clients, and they said, okay, we've got this technology, let's put it into an accelerated drug development. They pulled together a CTA and they went for it. And they came to us when they said, look, we've got some issues, can you help us? So what had happened is the agency that they'd submitted to had said, wait a minute, you've mentioned this bespoke cytokine, you provided a CFA. This really isn't enough. We need more information. We don't know if this is safe. This is completely bespoke. It's an entire material on its own. So they had a lot of expectations. So to counter this, we had to go in, we had to go back to even the supplier, provide more detailed specifications. And it was really difficult. We had to pull out a manufacturing description and some controls information. And there was so little stability data there, it had to be generated or mined from what they had historically. So this did result in actually significant delay and a resource review, and it was a critical approval issue, and it was eventually resolved. And so how do we mitigate this? I mean, looking back, as part of the standard risk assessment in the flowchart I mentioned, this needs to happen, this assessment against USP 1043 and EP 5212. These assessments need to happen. You need to have this information up front on the TSC, BSC adventitious agents. One of the other things that you might not have considered is to make sure that you have the right quality agreements with your suppliers, and make sure that you have contact not just to the person who sells you the material, but a regulatory contact who can reply to you very quickly. This is something that we really struggled with. The next example, and numerous agencies have come back to us on this one. So this is an example of many where human donor material information was insufficient regarding the controls for transport and storage. And we've had this for early phase and also even late phase, where I guess it was missed in the first review. And in this case, the agency said, how do you give us evidence that you've controlled this temperature controlled shipment of this donor material? And we didn't have the information readily available. It wasn't there. We could generate time. So we went back to the agencies who did accept our justification. We went in and we said, okay, well, we do use qualified data loggers and we are going to commit that they verified before we pre and post shipment and before we release this material for use into the process. And then because we didn't have any data, what we did was we gathered all the data from all the previous historical shipments we could, some of the development shipments, and we logged the shipment, the temp tail data, and we demonstrated in many cases they were shipments to testing labs, so we were able to say the product quality was maintained. It really wasn't the best approach, but it was all we had, and they did accept it, but they did restrict us to the conditions we had data on. So the widest conditions that we could mine from our data mining exercise were the ones we were restricted for our shipments of our material. So this was okay. We had to build the shipments for the clinical study around this. So a formal study was eventually planned and is being executed. So the mitigation strategy is to have that as part of all these assessments that you do up front, a complete list of all these materials, where they go, understand which ones are temperature labile and which ones you have to control shipments, and understand the quality attributes. So what you have to measure to show that their product quality is unaffected and therefore safety and efficacy. Another example, this one was about a manual process manufactured by a CMO that was then moved into a brand new in-house shiny plant from the manufacturer. They'd set up their own one, and it was fully automated closed procedure for an autologous therapy during phase 1. They used a CMO during phase 1, moving forward into the later stages, and the comparability data looked good, so this was looking great. However, and this strategy is normally welcomed going to the automated process with all these additional closed system. However, the downside of this, that upon their pre-inspection planning and readiness, they noted some pretty major compliance risks around the use of their computerized systems and communication of the system with their instruments. And this was a real lack of facility readiness, and the impact was quite severe actually, because they couldn't use the automated process. It was put on hold and delayed and actually planned as a post-approval activity, so they went forward with their manual process. Looking back now, I think we could have actually considered building this into a post-approval change management protocol and had that reviewed quite early on. A good mitigation for this, of course, is to build in the quality compliance side of the facility as part of the planning. Then my next examples, I can't go into all of them. We have this one, I think I'll go into, and then I will stop here, was for a viral vector seed for a gene therapy product, which was partway through the clinical program using animal origin free components, and a comparability assessment was required, and we weren't able to do the usual Biotech 3x3 pre and post comparison. Wasn't possible. They had very limited number of batches, and finished product testing actually was unlikely to pick up the impact of the change that we've made here. So a completely alternative approach was proposed. We asked if they could, if they would accept some development data and one post change data. And we included the in-process control results and some additional characterization. But because of the limited material, we really couldn't do so much. So after some dialogue with them, we included an additional in vivo testing to confirm that regardless of all this, with our safety data as well, that potency was also unaffected in an animal model. All of this together took over 20 weeks. It was a big piece of work, so it had a big impact. And we did get agreement on an alternative approach. So we weren't forced to go into an approach that wasn't feasible for us. They did work with us. You can't plan for everything as a mitigation, but if you identify those high risks, particularly of critical reagent materials and banks and seeds, this is something that you can have some backup strategies in place. I'll just mention process consistency, and this was my comment about trending your process on an ongoing basis and identifying your actual normal operating regions. We also had examples where potency method wasn't appropriately qualified early on. It was used over a number of years, and it was only very late it was identified to not actually be adequate. It wasn't fit for purpose. It wouldn't have actually informed us if the method had failed. So this all had to be repeated. We had to do additional crossovers. We needed retains from historical batches. I won't go into this too much. And then, of course, finally, a number of authorised products have been withdrawn. And we know this. The main reason cited for withdrawal was commercial reasons and manufacturing facility-related issues. So this is why I do emphasise the point regarding ensuring that your facilities are fit for purpose and that they're scalable. They're scalable and that the reagents and commercial requirements and the shelf lives are all commercially viable, including storage and shipments. So the last slide, you'll be happy to know, is the overview. So regardless of the product status, the CMC can't be skipped. And I'm sure it will be squeezed, but it can't be skipped. So some items can occur in parallel to review, provided on a rolling basis, but you still need to demonstrate you can produce safe product to the desired quality using a robust process. A balance between these accelerated timelines and risks is required with appropriate risk-based studies, and sufficient detail needs to be given to those post-approval supply logistics and any CMC facility issues. Risk analysis should be ongoing and proactive, and of course, approval will be based on a risk-benefit assessment, including the contribution of all the risks that come from CMC. So thank you. That's my presentation.
Thank you very much, Zeb, for this very detailed and very interesting presentation on the CMC aspects. Thank you, Yemi, for the non-clinical part. I want to tell you that we don't have questions from the audience at this point, but I would like to ask each one of you at least one question. We have 10 minutes foreseen for the Q&A session. And as my background is mainly regulatory, I wanted to ask Amy, what is the early regulatory support possibility that is available during assessment of the non-clinical studies before going into clinical trials with humans? Yemi, do you hear me?
Sorry, I forgot to unmute myself.
Yeah.
Yeah, so thank you for the question. The earliest really kind of depends on the region you're working in. So in the EU, there's something called the Innovations Task Force. And what that does is that looks at your early development. It looks at all aspects, but it's really important that part of the interaction with the regulators that you can discuss things like your preclinical and your non-clinical kind of plans, strategies. Look at whether the appropriate kind of models you're thinking of using are appropriate. Looking at whether the technologies, if it's like editing, gene editing, that would be the kind of safety aspects that you have to consider and what would be important. And I think also you could actually maybe start talking about the kind of information required for kind of safety aspects and maybe GMO. So that's the for Europe, for the EMA. For MHRA, it's the Innovation Office, which is fairly similar to the Intervention Task Force. And in the US for the FDA, it's called Interact, which was originally called the pre-IND. So again, that looks at the aspects of your preclinical data and clinical stuff and what have you. You have to be really important when you apply for these early interactions, because you can reach a stage where if you started your CMC or you started doing your kind of development, that you would be deemed to be more developed in your stage. So I know the FDA do that, especially for Interact, where they will look and say, well, you've established your development in an advanced stage, and so therefore they may not think that your questions would be relevant for Interact meeting, but will be moving towards a pre-IND stage. I think where the International Innovations Task Force, there's a cut-off point, but it's not as stark as it is for the FDA. But those are the kind of things you would look for. And now the FDA also used a committee called the RAC committee, which looks at molecular biological technologies. So some companies would have to go to the RAC committee, which is part of the FDA, to look at the new technology. And I know from previous experience that one company who was introducing a new gene editing technology used just that. And obviously the evidence from their non-clinical would actually move towards the basis of assessment.
Thank you very much, Yemi. After I very briefly marked the Innovation Task Force, thank you for giving us information on the other committees that can be contacted in this very, very early stage of the ATMP development. Thank you very much.
Thank you.
And I also have one question for Zeb. This is also partly regulatory connected, and it is related with applying for a scientific advice. And what would be your advice from CMC perspective? Which are the most important CMC topics and questions that is better to be addressed at this very early stage in phase 1, as you mentioned in your slide?
Okay, that's a really good question, actually. So I think it is something that I do think is often neglected. Scientific advice is planned and usually executed for a number of clinical, non-clinical questions, but I think it's really worth asking these CMC ones because it will prevent the questions during review, ideally to the region that you're applying to. And the kind of questions I would definitely focus on would be, are specifications acceptable? Do you accept our give them an outline of your process, ask them if the process stages and the level of controls and content you're providing fits in. And I know at that stage, you might not even have that information given how fast and dynamic these programs are. So even a commitment for what's going to be presented, and then ask them if they have any further comments. So I would say specifications, the manufacturing process and controls. And then with every one of these products, there's an issue. So whatever your issue is, if it is a particular virus you've identified, if it is a particular safety concern or a reagent that, for example, is bespoke, bring it up now and ask them, to let them know your approach to controlling this. So I would say definitely include CMC, if not on its own, then as part of the scientific advice, ask them to verify the specs and the release tests. In particular, of course, potency, your potency testing approach is acceptable, including your validation status. Ask them if your process and control general flow diagram is what they would expect, and then ask them about your specific issue that's specific to your program.
Thank you very much, Zeb, for emphasizing the importance of these CMC questions addressed so early, because really from regulatory perspective, often CMC and quality sections of the dossier are one that are most often challenged by authorities. So this is really important to be considered by the applicants. Now we have 5 more minutes, which I will add to the planned break. Thank you, Zeb. Thank you, Eva, very much for your presentation, for answering the questions, and for your participation. It was really interesting to me and to everybody else, I believe. We will be back in 20 minutes to proceed with the third and last part of our event today that will be dedicated to the post-marketing phase of the ATMP development. So thank you and see you in a while.
Bye-bye.
Bye. Hello, do you hear me? Welcome back. Hi, Annabelle. Welcome back after the short break we had. And now we are entering into the final third part of today's event, and we will be going through some of the ATMP post-approval aspects with regards to market access and access to patients. The topic will be presented by my colleague from PharmaLex Sweden, Annabelle Forsmark. Annabelle is a Senior Manager, Health Economics and Outcomes Research. Annabelle, please, the stage is yours.
Thank you, Miglena. Theodora, can you share my presentation?
Yes.
Thank you. And thank you for the opportunity to start this session about the post-approval path. So in a way, that is the most important part of the journey for ATMPs, since treating actual patients is the main purpose of developing ATMPs in the first place. Please. So the aim of the presentation is that I want to give an understanding a bit about the post-approval process and the ATMP specific challenges. So what is already very clear is that ATMPs have opened up possibilities to really significantly have a large impact and even saving patients' lives. So, and in the present situation, there is a number of ATMPs that have now reached marketing authorization in Europe. And some have already passed also through national processes, which are necessary for recommendation or adoption for market access. So once the market approval is in place, there is, of course, a natural strong demand and expectation from patients who don't really have the time on their side to wait for a long and inefficient process. However, unfortunately, marketing approval does not translate to market access, or even more importantly, it does not guarantee patient access. Actually, current structures for healthcare funding have not had time to adapt to ATMPs, and the systems for evaluating and funding ATMPs still lack the necessary framework for addressing these new technologies. So with this presentation, I would like to map out the post-approval path and to highlight the hurdles for ATMPs on the route to finally treating patients. Yes, please. So with this picture, I would like to illustrate again that the regulatory approval is not the end of the journey. So after a long preclinical and clinical regulatory process, which we have heard a lot about in the previous presentations, so this is a process of hard work and a lot of resources. So hopefully a positive risk-benefit assessment will result in marketing authorization. But then what are the direction for the next distance to cover? So by that I mean how does the ATMP actually reach the patients? Yes, please. Market access, when I say market access and patient, market access is the aspect of recommendation or adoption of use in the separate market, and patient access is actually treating the patients. And patient access is, of course, the very reason for developing the treatment in the first place. And most generally, an ATMP will really be a game changer for severe, often life-threatening disease, a chance of life that previously was not possible. So approval, once in place, will of course lead to very high expectations and hope for the affected patients, but also their families and the treating physicians. However, market approval marks the beginning of a process which actually is still very much in the making, and it's actually developing in parallel regarding ATMPs. So the first stop then is market access. So from the developing company perspective, this is a very important milestone. So market approval is really the starting point where there can be a potential return on very many years of investments. And there will be investors who, of course, are expecting that this will happen. With ATMPs, the technology often targets a very limited number of patients. So the investment refund must be based on a very small number of patients. But there is the same development cost as with the same development cost as for other treatment therapies. So that means a very high cost per patient to return the investment. Many times the companies behind the ATMPs are small biotech companies that are built around a single ATMP first-in-class technology, which have no other marketed products to cover the periods ahead of commercialization, which can be a very long process. So it's a matter of make it or break it. So in order to gain market access, countries evaluate different sets of criteria for recommending or implementing a treatment. That means that there will be separate national processes that are really resource consuming, and that might mean that companies do not prioritize smaller markets where a limited refund is expected. This in turn can lead to a very variable pattern of access. And this is of course a very undesirable situation when it comes to these potentially life-saving technologies. You don't want to have a variability in access, of course. Yes. So next I would like to describe a bit the process then following the marketing approval. So generally, as for other therapies, ATMPs will have to pass through a process of a health technology assessment, or an HTA in short, in order to have a recommendation of use or no use. And most countries have their own HTA bodies who are responsible for providing evaluations for informed decision making. And that is of course crucial because, well, essentially all countries will have a limited defined healthcare budget that must fit all available treatments. So it's a matter of prioritization. So the recommendations from the HTA bodies is a matter of, it's important for the prioritization within this limited budget. So the methods and priority of different evaluation criteria for making these HTA evaluations are different across different countries. So focus might be put on, for instance, unmet medical need, in that assessing what treatments are already currently available for the patient group. So it might be that an ATMP targets a patient group that already has a treatment which is maybe invades life to a large degree, but having the treatment makes the patient group not well. It's not patients that are they have a treatment that will make them go, even if it's an invasive treatment. So the unmet medical need is of course something that is looked upon. And also, of course, the therapeutic value, the magnitude of impact on the condition and the patients. Another criteria is, of course, the value for money. So what is the benefit achieved at what cost? And also the total budget impact. But it might be hard to, when hearing all these fantastic possibilities with ATMPs, it might be hard to understand why the road isn't really wide open for ATMPs, because the medical need is often huge. The treatments are often potentially curative. There are very innovative technologies, and which should be supported according to many official directives. Okay. There are, of course, always challenges with market access for many treatments, but there are roadblocks in the route from market to patient that put ATMPs in a very specific league on their own. And the first point is, of course, what we've already heard several times to be repeated, is the price tag. So ATMPs generally come with a one-off very high upfront cost, so it is a matter of affordability. There is often also very large budget impact, which budget impact is the patients times the cost of the treatment. So despite very few patients, the budget impact can still be huge due to the cost of the treatment. The second point, which we have already also touched upon in previous presentation, is the point of uncertainty. So in general, the level of evidence is much lower than for other treatments. So the use of, for instance, surrogate endpoints for survival and morbidity, the extrapolation of effectiveness to a lifetime horizon from just a few years of clinical trial data. Also, for natural reason, the trials are open label, non-randomized trials, which also enhances the degree of uncertainty. There is also an important point of future technology development that is also often raised. So payers will be paying a very high sum for to buy an expected lifetime effect for the individual patients. but it's really hard to know how the scene will look like in the future. So as we also have heard, there are competitors in the pipeline and also other technologies. So there is a very high upfront payment for something that might cost very much less in the near future. So the third aspect with ATMPs is also the requirement for healthcare organizational changes. So it's a question of readiness. So most often ATMPs will be offered in qualified centers to enhance the expertise and also to handle the infrastructure associated with transporting of cells and patient material. There is also what separates ATMPs from other therapies is an additional cost for the healthcare. So it's not just the cost of the actual therapy, but there are a lot of costs for healthcare, considerable cost for healthcare regarding pre-treatments, hospitalization, and also later complications that must be borne by healthcare, so in addition to the already high cost of the therapy. And the last point concerns the current HTA systems. So HTMPs, due to their nature, simply do not fit into the current framework. So there are no good models to really capture the long-term costs and benefit of ATMPs. Yes. So if elaborating a bit on the specific HTA challenges, so again, importantly to remember is that HTAs are used for informed priority decisions in healthcare. So the outcomes of the HTA assessments will form the basis for recommendation of use or no use. So as already pointed out, there is a limited budget, some to include all different kinds of treatments. So some countries translate clinical outcome to generic measures like quality adjusted life years or QALYs. This aims at fitting all the treatments into the concept of receiving the maximum amount of health for the money spent. So generic measures shortly enable to compare the value of very variable treatments, all from ATMPs down to, for instance, migraine medicine. The problems arise from an HTA perspective, the problem arises when there is a very good value for the money, but the treatment still doesn't fit into the budget. So actually many ATMPs have a very compelling cost-benefit ratio. The cost is of course very high, but the benefit is large. And if there is a system using a cost-benefit ratio for recommendations, so why are ATMPs not easily adopted if they are such a good value for money? That can be explained by a health economic term called opportunity cost. So shortly, that regards the opportunities that are lost if implementing the technology. So something within a limited healthcare budget, something else has to be pushed back. So this is concerned with any high-cost treatments. So theoretically, a few ATMPs could push a range of other treatments out if being evaluated within the same system and budget. So a measure to address this problem is to have dedicated budget for specialized high-cost therapies. One example are the UK Cancer Drug Fund, for instance. So the current status and concerns. So end of 2019, there were currently 10 ATMPs who had marketing authorization in Europe, and 4 had been withdrawn for commercial issues. And and we already heard that from some of the other speakers, and that it is quite remarkable to regarding the very long and costly path of development to take the therapy out of the market for commercial issues. So obviously there is a large challenge to commercialize these technologies. And also, as we've heard in early presentations, there is a very large number, several hundreds in the pipeline, somewhere between phase 1 and 3. So even if the few available ATMPs today can fit into a country budget, affordability will be a concern for most countries in the long run. There is also the payer aspect. So if a positive recommendation, who will actually fund the treatment? This can be troublesome in the case of many endemic or regional conditions, so where patient numbers can be very variable across countries and even regions. So actually in Sweden, this is a current case where there is a central decision, positive recommendation of use for treatment based on a beneficial cost-benefit ratio, but then the patients are actually concentrated to, well, mainly concentrated to one region in Sweden, who are then supposed to fund the treatment based on the positive central recommendation. So there is also some conditions that are very variable in prevalence. For example, thalassemia, which is very highly prevalent in some countries and only have a few patients in other countries. So patient access can be very feasible in one country, but then might be a very long and hard process in another country. And that, of course, leads to the very important aspect of equal patient access. So if there are different recommendations in different countries, that is, of course, of a very special concern when it regards life-saving treatments. So there are actually patients actually move abroad with their families when this situation occurs, when it concerns these important therapies. Also, many of the ATMPs are actually intended to be, because of this high variability in patient numbers, and also since you want to concentrate the expertise and also to have a feasible logistic around patient materials. So most often ATMPs are intended to be delivered at international treatment centers and not offered in each country. So that also leads to special considerations regarding the patient resident country budget, in the sense that national priority decisions might be bypassed. So what is done to address these challenges? So there are a lot of initiatives to address not only ATMPs, but also high profile or costly therapies in general. So there is a trend towards joint assessments. So there are a number of collaboration initiated in Europe for joint assessments, for example, between Belgium, Netherlands, Luxembourg, Austria, and Ireland. There is the FINOSI process, which involves Finland, Norway, and Sweden to make joint assessments for new therapies. And also UNETA, which includes HTA representatives from many countries and who provide a framework for HTAs, which then must be adapted to the national processes. There is also initiatives for joint negotiations. So the FINOSA, there is a recent decision within this FINOSA initiative after a joint assessment within FINOSA. The respective countries and Denmark have taken the collaboration further in making a joint negotiation for the treatment. And also that was the case for Spinraza, where Belgium and Netherlands had a joint agreement for Spinraza, joint negotiation and agreement. There are also payment models that are in development. So these are designed to address the high one-off cost and the high degree of uncertainty regarding this long-term effect. So there are a range of payment models that have been proposed. So there is conditional reimbursement, which is associated with a condition for follow-up with real-world evidence data, and then to assess the reimbursement again after a period of time. It has also been proposed to have annuitized payments that are spread over several years, because most often such a high price tag will not fit into an annual budget in healthcare. So they also pay for performance-based agreements. These are based on treatment success and include some form of refund if there is no successful outcome of the treatment. And there might be also combinations of these payment models. However, it's most often the implementation of these payment models is quite challenging because you have to set and agree and define the criteria for the agreement, and also to have adequate routines to follow up the agreements. So I would like to use the CAR-Ts to illustrate how these challenges that have been described have impacted on the adoption of ATMPs. So the CAR-Ts were the first approved gene therapies back in 2017, and they are immune therapies with the patient's own white blood cells who are genetically modified and then reintroduced to the patient to attack tumor cells in special forms of lymphoma or leukemia. So due to the high cost and large degree of uncertainty, ATMPs are often marketed with a very narrow indication and often for use as second or even subsequent lines of treatment. And the uncertainty is often addressed by cost agreements. So consequently, the current recommendation for CAR-Ts concerns second or subsequent lines for refractory or recurrent disease. And the recommendation of use is in general based on confidential agreements regarding refunding or cost containment. So the chart in this slide uses the Nordic countries to visualize how the adoption looks for the CAR-T therapies Kymriah and Yescarta regarding in the Nordic across countries. So the first aspect to look within country differences. So you have, if looking at Sweden, on the indication of lymphoma, so Yescarta has been recommended, while Kymriah has no recommendation for the same indication. And the second aspect is across country differences. So if looking at a specific indication and therapy, for instance, Kymriah in lymphoma or Yescarta in lymphoma, so the recommendation of use varies across the countries for different reasons, either because of the price tag or for the level of evidence. So regarding Kymriah for the treatment of leukemia, the level of evidence has been found and the cost has been found acceptable and is adopted in all Nordic countries. However, in Denmark, there is a condition to collect real-world evidence data for a follow-up period of 2 years. So this illustrates how it looks for CAR-Ts, depending on these ATMP-specific challenges. Yes? So some concluding remarks. So ATMPs, the aim of the presentation was to highlight a bit. I think it's very easy to, when looking at these fantastic therapies, what comes to mind is why are all of these therapies not easily adopted? But there are specific challenges with the cost and funding and opportunity cost for other treatments. There is a high uncertainty of effect. It's very hard for HTA bodies to know what they are paying for. They're also in opposition to, that differs them from other non-ATMP therapies is the additional healthcare resources that are required. And also, of course, the lack of prioritization framework to assess these therapies. So, as was seen in the previous slide, the access to CAR-T therapies is already highly variable, and there are a large number of ATMPs, and many of these have a much higher price tag than the CAR-Ts. So, but still, the impact on ATMPs for the individual patient is huge. I mean, these are often curative treatments. So there is really an urgent need for system to address these challenges. And those solutions must be acceptable to both the payers and the companies. So I think Violeta stated a good point in her presentation that innovative technologies require innovative funding models. And lastly, I'd just like to quote an ecologist at a conference which focused on HTAs and ATMPs. And he concluded his talk by saying, why are everybody just focusing on the challenges? Why are we not just focusing on the enormous potential with these therapies? If there is a will, there is a way. And I think we will hear the most important perspective, the ones from the patients, in the next presentation. So really looking forward to that. Thank you.
Do you hear me now? Yes, I think so. Thank you very much, Annabelle, for your great presentation and giving us the insights of the reimbursement perspectives and for paying special attention to the CAR-Ts, because this is really a great transition, in addition to what you said last, to our next participant, Mr. Jonathan Clark from Jonathan Clark & Partners. He will give us some first-hand experience as a patient, and he will tell us about riding the clinical trial roller coaster. So, Mr. Jonathan, please, the stage is yours.
Thank you very much. Can you hear me?
Yes.
Good. Okay. Well, thank you, Annabelle, for setting me up wonderfully. And you had some very memorable phrases in there. I think chance of life was a good one. I'll come back to that. But before I start, I'd like to thank the organisers, PharmaLex, for the invitation to speak. It's really an honour and a pleasure. And particularly, I'd like to thank Peter Kapitein, who was the bridge between my hematologist and the organisers, and of course Miglena for all her work in setting all of this up. So thank you very much for that, and I'm really delighted to speak. Let me start. Hang on, didn't work. Yeah, let me start by introducing myself. The patient. This is the patient view. I am the patient. 2014, I was diagnosed with mantle cell lymphoma. I was 52 years old at the time, which is quite young for it. It's a very rare disease. It's unfortunately, so lymphoma is one of the success stories, I think, in recent years. What used to be an absolute death sentence has become very much treatable, and people are talking indeed of curable, and some forms are. Unfortunately, mantle cell lymphoma is one of those stubborn variants that is resisting efforts, at least up until now. I had chemotherapy followed by autologous stem cell transplant. That's the protocol for mantle cell lymphoma, and it appeared to work. I was sort of hoping for 8 to 10 years. That's the median, 8 to 10 years of remission. I knew it would come back. It always does. Unfortunately, I was unlucky, and it came back very quickly. In fact, it possibly never went away. So we'll call it refractory, relapsed, whatever it was. I'll come back to what that means, but it's basically not a good thing. I had, and this is actually kind of interesting if you think about it, ibrutinib had within months, had been approved for use in the European Union. And I was lucky enough to have that, and it did a good job for a while. Unfortunately, not for too long. And the main body, what I'd like to talk to you about today is really how I came to be in a trial, what it was like being in the trial. But I wanted to give you this sort of background information. And of course, the story is that I'm in complete remission 2 years later. It's remarkable. When I think back, I really shouldn't be alive. I should not be here talking to you. If I lived in another country other than the Netherlands, if I lived, or the US, if I lived in a different city in the Netherlands even, if I was born 6 months, a year earlier, I wouldn't be here now. The only reason I'm here is the clinical trial. And that's sort of the underpinning of this. And I promised myself when I was lying in my hospital bed that if I made it through this, I would try and tell my story to as many people as possible, to patients, to healthcare professionals as yourselves and the participants here, but really wide story, because I think it's a story that needs to be told as widely as possible. So that's me, Jonathan Clark, the patient. Just a quick introduction. Most of you will know, I'm sure, mantle cell lymphoma. Most articles, most scientific articles start off with this wonderful phrase. It's either dismal prognosis or associated with a dismal prognosis. It is pretty dismal. When it relapses or is refractory, it really is bad, and response rates are low. The overall survival of less than 3 years. Ibrutinib has helped, but if you stop responding to ibrutinib, which I did, the prognosis was bleak. Basically, what I was facing, I had too much disease to have another stem cell transplant, even though there was a donor. My only option was salvage chemotherapy, which I've never had, but I'm sure it's not a good thing. So that's the background. Why is that not? Yeah, there you go. So what I wanted to really do is dive in, and please feel free in the Q&A to ask me more details about my up and down journey through all of the different aspects. But what I wanted to do, because I talked a little bit about that yesterday, what I wanted to do today was to really focus on the trial aspects and try and it's a bit difficult. So I'm not quite sure who's the audience, but this is really from my perspective. How did I discover about the clinical trial? Well, I mentioned ibrutinib. I had a colleague and friend who has a form of leukemia, and he had been just recently put in the United States. He'd recently been admitted to a trial for a color in ibrutinib, which is the second generation of the ibrutinib drug. And he was saying, "Oh, you want to look for a trial? It's much better and it's more effective, less side effects." And his wife works for the NIH. And so she did some searches for me and found clinical trials, introduced me to clinicaltrials.gov and the register in the EU. So I think one part of my discovery was myself as an active patient. Once upon a time, I used to be a scientist, and I have that kind of scientific curiosity and skepticism not to trust everything I read on Google. But even so, that patient-to-patient network of, "Oh, I heard this, and did you know that?" And my wife, and "Oh, I have a friend who," is very powerful for us, especially in this situation. You know, there aren't too many options. This isn't, it's not like it's hard to really describe how black things look when you've given a diagnosis like this. So you grasp at anything. So that's one angle, and I discovered myself. I saw that the trials for mantle cell lymphoma in the States, in China, just opening up. And so I was about to go to my doctor and say, "Hey, how about a trial?" when she had also done her work in the background. And as it happened, her professor had just been contacted to take part in the trial. And so through these two networks, we ended up identifying a trial for my disease. Now, what follows, I have no idea if it's typical. I've only been involved in one. I sort of pick up here and there that these kind of things do happen. And I did some detail just to give you a feel for it. So in February 2018, that was the first possible mention of a trial. I found it. We were interested. I said I was interested. But unfortunately, in March, the trial closed because there were some unfortunate deaths of patients, and they closed it for a moment. Thankfully, they reopened it. So I said, yes, I'm still interested. The initial screening was done and I was included. I then had a blood test which showed some abnormalities, and so I was excluded. I had more blood tests and scans and was in and then excluded, and then, oh, I've missed off a detail here. I was then included again. My cells were taken out of me, leukapheresis, or as Annabelle mentioned, it's white blood cells, the T cells in particular, taken out of my body, genetically manipulated, and then you wait. And it's called bridging. I had to come off the ibrutinib. The question was, would I survive? Obviously I did. But in that period, I had multiple blood tests and scans. I was in again, I was out again, I was in again. And in fact, it all went up the very night before I was due to have the treatment, due to have my cells put back in. The final blood test showed that my oh, no, I've forgotten the name of it. It's gone. One of my indicators was too high, and so I was out, excluded the day before my drugs were about to come in. I got in, woke up the next morning, or went into hospital, and heard that they'd spent most of the night analysing and decided that the high level was due to stress, which was hardly surprising, and that I was back in the trial. And then a final blood test came back just 5 minutes before the central line was put in. Now, I think this is quite dramatic. I don't think it's necessarily normal, but I do think this up and down of being included and excluded is a particular part. And of course, it's for safety. I mean, they're excluding me because they fear that the treatment will kill me, not because of any other reason. But even so, as a patient, you have this roller coaster, which is why, of course, I've used the roller coaster image. This roller coaster of and when the bottom of the roller coaster is basically salvage chemotherapy and palliative care, it's a rough ride. But just as Annabelle said, the chance of life, the chance of a second life, this also is quite hard to explain to people that haven't experienced it. And I do so hope none of you ever do experience this. But I feel like I'm being given a second life. I should have died, should not be here, but I am. And I'm completely focused on living my life to the best as I possibly can, to tell my story, to help other patients, to do whatever it takes. I've been interviewed. I'm doing some videos for people just to try and help. I go back to the hospital just to show people what this kind of treatment can do. It's life-changing, it's life-saving, but it's life-changing as well. And I wanted to share with you my mantra. I love this quote for a long time, way before I was ill. It means an awful lot to me right now. So that's sort of my story. What I wanted to share with you are a couple of observations and two pieces that I think perhaps, or at least as far as I've seen, don't quite get as much coverage as they might. The first one is uncertainty, and of course it's clear from you, but I think this is inherent in very many clinical trials. By definition, this is innovation, it's cutting edge, it's advanced therapy, it's new. It's new not just for the patient, but it's for the doctors, for the healthcare professionals, for the pharma companies, for the regulators. It's completely new and innovative. And so it brings with it, by definition, a very high level of uncertainty. And actually, at times, just like the wonderful quote from Ursula Le Guin, it feels intolerable. How can you tolerate this uncertainty? How could I sit there in the hospital 5 minutes before I was due to go in for the treatment and be told, we're not sure you can have the treatment? But you do. You get through it. You deal with it. Of course, good old Dr. Google can't have much either because there's not much known. So if you go search, there's not much there. There aren't many other patients to ask. You know, right in the beginning, I said it's patient-to-patient network is strong, but if you're on a cutting-edge trial, I was the first patient in Europe with mantle cell lymphoma to have CAR T cell. So on this trial, so yeah, who else would I ask except myself? And so your questions go unanswered. Right now, my T cells are still supercharged. I think there are CAR T cells still in my body. They've depleted my B cells, so my immune system, my antibody system is pretty much shot, which means I have monthly transfusions. Some patients don't need that anymore. Some do. Why? We just don't know. And you're so used to going to doctors and health, asking the questions, and they give you an answer. And now I go, and of course, they say, well, that's a really good question, Jonathan. That's an excellent question. We're not sure. Here's some theories. Example, am I particularly vulnerable to COVID? Well, maybe, but maybe not. We don't really know. And that level of uncertainty, if I was talking to a patient that was about to go on a clinical trial, this is what I would be talking to them about, how to deal with uncertainty. And this is my current answer. Another quote, I think you need hope. And in fact, when I was doing a bit of research before I was due to speak here, I discovered there's just a few papers on something someone has defined as therapeutic optimism, which I really rather liked. You just need optimism and hope to cope with that uncertainty, because you need to see the roller coaster in front of you. You need to see the upswing. You need to know the duration of life in order to cope with as you go down into the pit. And actually, I begin to wonder, especially thinking of the wonderful staff, the doctors and the nurses and the healthcare professionals and the physiotherapists and the nutritional experts that help me. I wonder if it's just as much as important for the staff as for the patients, and perhaps also for you all involved so deeply in clinical trials. Maybe some therapeutic optimism is good for all of us. And I love that piece at the end from Annabelle, the quote. Yeah, I think so. Maybe we need to focus more on this, because it is difficult. You know, you could be too optimistic. There's a balance. I mean, obviously, physicians, doctors have a do no harm mantra, and you need to balance that with optimism. The best way I can describe that is a little anecdote from my treatment. So I had my cells, they went to work. I had a huge cytokine release syndrome, enormous cell death. I was put into intensive care because my blood pressure went down dramatically. I came out of intensive care and things looked okay. And they managed to control the blood pressure with other means, because they were holding off giving me steroids, because give them too early and the cancer might have a chance to come back. Give it too late, I'm going to die. So that's the choice that my my carer had, that my doctor had. When do I give Jonathan this? And in the end, it happened. I had a neurotoxic event, but all I could tell him was, I don't feel right. So on the basis, this is all I could say. He asked, but all I could say is, something's not wrong, doctor. Something's wrong. Something's not right. I don't feel like myself. And based on that information, he made his decision. And I think back a lot onto how that particular doctor acted, and how he balanced his optimism that the patient would live with his need to do no harm and to issue me with the steroids that would stop that would decrease the cytokine release syndrome. So it isn't easy, but I'm living proof that when you get it right, you give someone a second life. So I want to thank you. I can't thank all of you enough for all the hard work and the effort you put into clinical trials. I've just had one variant, but there are many out there. There are many more patients like me. There are some, sadly, who don't make it, but there are many that do because of what you do. You've all helped to give me and everyone else a second life, and that means a lot. Thank you.
Thank you very much, Jonathan, for sharing that experience, which is, first of all, it's very nice that you went through it and you survived it. It's a perfect outcome, the desired outcome. This is actually the aim of the therapies. And thank you for sharing this, because this is very, very personal. And I think that this is the side of the ATMPs and all the challenges, that this is not put out there too much enough. I would say it's not put out there enough. So thank you for doing that for us today at our event.
Pleasure.
Annabelle, thank you too for your presentation and giving us the perspectives of the reimbursement. We are quite ahead we have quite some time left. So I will ask each one of you a couple of questions, and I would encourage if any of the other participants would like to say something or ask you a question to also do so afterwards. Annabelle, you mentioned that often for reimbursement reasons, patients from different countries, they're forced to make the hard decision to change their residency so they can have access actually to ATMP and fight this battle that Jonathan just told us about for their life. So how do you think this unequal patient access to ATMPs can actually be addressed?
I think it's a very crucial question, because I think, I mean, it's always unequal access to healthcare is always a very important aspect. But I think especially when it comes to these life-saving therapies, I mean, it's a very undesirable situation to have unequal access to these therapies. So I think there is really a need for to take a centralized approach. So I think one solution could be, because of course, I mean, it's understandable from the company's perspective that they need a refund from the long development programs. At the same time, it's really understandable from the healthcare payers that, I mean, it's simply, as we saw all these ATMPs coming along, there is simply the affordability is just will be a real problem. So I think I know the ATMP dedicated specialist group in Sweden have worked with, where both industry and academia and government are working together. Is one proposal was to have a good set of framework and transparent, a good and transparent framework, so you can communicate to companies that they don't have unrealistic expectations of refund. So one suggestion which I thought was very good was to have different payment models for different target product profiles. So you have a number of classifications of ATMPs depending on the effect, the patient group, well, different aspects. So, and then you have a transparent way of communicating to developing companies that this is the framework that you can work within. This is the cost that you can that is reasonable to expect. So I think that is one way of approaching the very high cost for the to somehow to have a transparent communication between payers and companies that are acceptable to both. And I think in the long run, I think there is really with all, because when you see the pipeline with all these new therapies coming along, I think it's really needed a kind of solidarity funding, maybe on a European level even, because there will be countries with less opportunities to pay for these treatments. So I think it's really a solidaric funding of these therapies, maybe through a very central body on even EU level, could be also a solution in the long run. But I think, I mean, as I think it was Violeta who said, I mean, these are, and also as you so vividly illustrate, Jonathan, I mean, these are fantastic technologies. They save lives. I mean, there is somehow it needs to be addressed. The technology is here. I mean, it's no longer a matter of choosing or not. I mean, the technology is here, and it's here to stay. It's even coming along in more numbers. So there is an urgent need to address it somehow, and really to ensure equal patient access. That's a really important issue.
Yes, definitely. This is crucial for the actual use of the therapies and them allowing people to be healthy and live long, happy lives. And the topic you addressed, that communication and discussion between the governments that are actually funding those therapies on this solidarity model that you refer to, and the ATMP developers is really, really very important. But do you think that if this model that you told you find very good approach currently to put some ranges and communicate that with ATMP developers, that what they can expect as refunding ranges and possibilities? Can this lead to a situation where some ATMP developers actually make the hard decision to cease the development and stop the product?
It's of course a risk with that kind of model that some ATMPs will not fit into those ranges. At the same time, I think there is some need for some more formal framework around that, because I think it's also hard for the developing companies to have a range of investors, and there are unrealistic expectations maybe for the refund. So I think that is the next. It's a very important issue that you raise, Miglena. So that will be the next issue. But first and foremost, I think you have to take a formal approach to somehow regulate the scene for ATMPs in some way, because I think the cost tags are really escalating, and I think also in the long run, I mean, the molecular background to the therapy, I mean, it will be, as for most technologies, in the long run, there will be more of a routine, maybe technology, and the prices can come down. So I think, I mean, there's so much uncertainty still. I mean, hopefully how it will work with all the logistics around transporting patient material. Everything is so new, and it's really hard to see how the treatments are going to, and how the process around the treatments are going to work ahead.
Yeah. Yes, definitely. But that what you said, that in the long run we can hope, and if we're lucky, actually see some cost decrease, that would be very, very beneficial to the healthcare care systems, and of course, most importantly, to the patients.
And just take the opportunity to thank Jonathan also too, because I'm working with HTAs, and most often you end up in a very theoretical discussion about generic measures of quality of life and costs and cost-benefit ratios, and in all those discussions, the actual patients are almost forgotten. So I think it's so important to get the faces to really have a reminder what the discussions are all about, actually, and to really have a constructive approach to addressing the challenges.
Yeah, thank you. I was going to say I'd be more than happy if anyone on the call at all would like to contact me. You have my details, or through Miglena. I'm more than happy to tell this story as widely as you wish, because I think there's some important. There really are some important things here. I mean, you see, I was thinking, as you said, about, I don't know if you've seen it, but some patients, I've seen crowdfunding efforts. People are, their country doesn't do it, so they've gone out to their family and friends and onto the internet to crowdfund the money to fly to America to have therapy. And somebody asked me about that, and I have real, maybe it's because I've grown up in Europe, but I have real ethical, moral problems about asking family and friends for money to save my life. But these things, it shows you how desperate people get and how hard it is. And I agree with you. I think some kind of framework to help is, because this is good news. This is seriously good news. I mean, I'm alive and I'm loving it. And I every moment and there are, 68% of the people on the trial are like me. They're in remission, complete remission. It's a stunning result. And I know that, so I can't tell you which trial I was in, because it's currently under approval process at the European agency. But it's going to come somewhere soon. And the other thing I was thinking, mantle cell lymphoma is relatively rare. And so actually to build up experience, if you're a center, a hospital wanting to offer CAR T-cell therapy, where do you get your experience from? And so there's another reason, I think, for perhaps a European framework, so that healthcare providers in different countries can gain knowledge from each other, because really the genetic manipulation is actually only part of it. That's the stuff that goes in your body, but the care, maybe I tried to say, but the care after that and my recovery and knowing when to dampen down the cytokine release syndrome. This is a dedicated group of doctors and nurses. I had, instead of the normal situation where people come on and off and they're on call, off call, I had a dedicated group because they had to understand my mental state so that they knew when I had a neurotoxic event. So this is a really different way of healthcare as well. And I think that is also just as important almost as the development of the drug, of the treatment.
Yes, definitely. It sounds like a very important part to make the treatment process actually successful to the very end. Yes, Zaklina, please.
That reminds me, Jonathan, what you just said on the care after recovery reminds me of something I heard in one of our last committees of the pharmaceutical the German committee. So we were discussing so this aspect of centralization. So for treatment, and then what happens after the treatment. And so that there, these concepts, okay, that the treatment will be centralized for very specific, very experienced clinics, but that recovery, so the treatment after the recovery, the follow-up should also happen in more clinics, so that also the patients have the chance to, well, not have to move always to a far away clinic or something like that. So that is currently also in Germany in discussion with regard also to reimbursement and how much money, which clinics are allowed to use CAR-Ts, etc. So yeah, thank you.
Yeah, I mean, oh, my pleasure. Yeah, I think it is. When I had my first autologous stem cell transplant, that was in Amsterdam at one of the centers in the Netherlands, but my recovery was at my local hospital where I live. And that was the first time, that was the beginning of a program to push out the recovery phase to regional hospitals. I think it's absolutely a good idea. Wasn't entirely successful I think there are subtleties involved in treating patients with depleted immune systems and guarding against infections that are second nature to experts and are not so obvious for new things. So, but I think what's really powerful there is using virtual techniques. I mean, there's so much now on video linking. I think you can connect people to experts much more easily now. So I think that's probably a way to mitigate that effect. But I have now, interestingly, one of the breakthroughs in this country, I have monthly immunoglobulin transfusions, which 2 years ago I had to go to the hospital for that, or a year and a half, but now I have home care. So a nurse comes to hook me up, all the equipment's in my house. He comes to hook me up and I have the transfusion here in my home, and that on quality of life, I mean, it's a win-win. It saves money. It means I don't have to go to the hospital, so it's safer, and I love it at home. It's win on every front. So I really like this kind of thinking in modern healthcare. I think it really is patient-centric, and it's from my perspective works very well.
Thank you both, Zaklina and Jonathan, for this point. And Jonathan, you're totally right about mentioning that now having all those means of communication and conferences and all the various platforms, it's very easy to connect patients and healthcare professionals in different hospitals that have to take over the follow-up treatment and recovery. And I would personally say that thanks to COVID, we now realize that more than ever, and we are using it more than ever. And having this event virtual online is actually a very good example of that, because if it wasn't for the global pandemic, probably we would prefer to have it live. But this gave us another perspective and a very, very good opportunity to connect people all over the world on various topics and ideas and discussions without leaving the comfort of their own home, as you said. And I also would like to ask you one question. When you shared your experience that you were at the hospital waiting for the treatment, and 5 minutes before that you were told that this is not 100% sure that you can get it, I guess that was very bad 5 minutes of the whole process. And as a patient, how would you address that and recommend how this could be improved? So such situation for people who are in the position of the patient, who are emotionally very fragile and not in a good place to not be put through the situation.
Oh, that's a good question. Well, I think, so I totally understand why, the exclusion criteria are, well, it's partly, because I read the trial protocol, so some of the exclusion criteria are for scientific reasons. Most of them are for safety, at least in the trial that I was in. And so I understood it. That's kind of what I meant by, I would like a little bit more understanding and perhaps management of the uncertainty and the hope. I don't know. I would have liked to have been able to talk to someone who's told me a little bit of what I might expect. I faced it. It happened. I think there was, on the one hand, from the doctors, perhaps a little bit too optimistic. They were saying, "No, it'll be fine. Don't worry, it'll be fine." And I would personally have liked someone to say, this will be a very difficult roller coaster because it's important, and just to sit down and explain it to me. And yes, I have a document I signed which said that in legal, the regulatory people did their job. It is written that things could go, but not approach it from the non-medical side, more the psychology of how it is to be in a trial, how to deal with uncertainty, the role of optimism and hope, and how your family can help. And it's not just me. People would call me up and say, well, why is that? And it's quite hard to tell people that you don't know and the doctors don't know. It doesn't give you confidence. But if you understand the process, then I think you can explain it better. And so I think more communication and more appreciation of the psychological aspects of it, that would be nice. I think we tend, because it's a hospital setting, there's a very much a feeling of, if you're sick, we'll give you a doctor, but not preemptive. So if I'd been psychotic or I'd been, I'd really gone crazy, I'm sure we would have found a psychiatrist, psychologist to help. But I was totally fine. Still would have been nice to talk to someone, it just to prevent, and it wouldn't have changed anything, but I would have been a bit more balanced about it than I was. I'm not sure if that's a good answer to you, but that's really why I picked out those two elements. I also, what I talked about yesterday on quality of life, I think things like food tends to get, with sick patients in a hospital, you get better. I think there's a deeper level of healthcare which looks at, you're a healthy patient, how can I make you more healthy? Or you're fine, but can I help you deal with what's going to happen? Preemptive in a way. Tough call, but I'd love to see that. I'd like to help.
Thank you for that answer. It actually answers my questions very, very well, and it raises another question by me. Do you think that it would be really beneficial to the patients if this is considered by the CROs conducting the trials to assign also a psychologist who should talk to the patients, as you said, and explain to them not only the legal part and the medicinal part that they already have been familiar with, but also the human and emotional part and what they can expect, to know that they have someone who can they reach out to when they're feeling down and uncertain and concerned, and all of the feelings that the patient can feel.
Yeah, I absolutely agree. But I think it's the same. So I'll give you an example on the food side of things. So, hospital food is, well, anyway, these treatments mess your taste buds around. Hospital food isn't great either. And so I just didn't eat much. And instead of, what they send is a nutritionist who says, well, you're not eating enough, here's a diet. And that really doesn't help, because I'm not sick. It's not that I'm not, it's something else. I would, the correct answer to that for me was, here's a personal chef who'll prepare some wonderful dishes for you. Okay, that's a bit over the top, but the same thing. I worry in a hospital setting, if they assign a psychologist, it's because I'm somehow ill psychologically. And this is something different. It's more like an advisor. It's more like a trial advisor who is understanding of the psychological aspects, but is supporting you. I had a physiotherapist who was just a wonderful, wonderful young woman. And all she wanted to do was to get us up and moving at some house. So, you know, we're all various people with various cancers, mostly leukemia and lymphoma, all of us in different stages. But she just was infectious in her enthusiasm to get us up and moving. And she wasn't treating us. She was making us feel better about ourselves. And I think that's the trick. Is there someone who can who can build our hope and our optimism? So help us on the upside, not just on the down. Does that make sense?
Yes, it totally makes sense to me. I hope to everybody else. I don't have any more questions. If somebody else has, please feel free to ask them. Obviously not, so I want to thank you all, and to close the event, thank you. Oh, Zaklina, please go ahead.
Okay, there's some time left. I was just thinking if I can ask something more to Annabelle.
Of course, please.
Well, I don't know, Annabelle, you mentioned, of course, that the health technology agencies are requesting real-world evidence. I mean, this is a huge topic, of course, and you only were able to touch this very shortly. So I don't know if there's maybe 1, 2, 3 more sentences you would like to add, what could be improved, or what the applicants or the marketing authorization holder should take care of, maybe also earlier in the development.
It's a very good point. Yes, so time didn't allow to go into that, but it's as you say, it's a very hot topic, especially regarding the ATMPs. So, and much of that comes from the, of course, uncertainty regarding the clinical evidence because of these non-randomized open-label trials and the short trial period. So there is really an urge to see how it looks like, how the clinical effect is, and what it looks like in clinical practice. So I think that, and most often I think there will be frequent demand of to follow up with real-world evidence. So I think companies can be very proactive in already addressing and to proactively collect what elements would need to be substantiated from an HTA perspective, and to already, when submitting HTA dossier, to already have a plan for collecting real-world evidence data, because most often that can also form the basis to have an agreement with the payers to already have a good collection of real-world evidence plan. So I think companies can really be, it can be very valuable to already have that in place. And also, so also to already to consider what, from what sources one might be able to have the real-world evidence. So to have a plan for, is it collection by chart reviews or registries, or to really have a plan and a design. I think that is a very good proactive work that can be started quite early on when already acknowledging what the challenges will be regarding to substantiating the clinical benefit.
Thank you, Annabelle. Maybe I can also add from the regulatory perspective. So I think I said in the morning, and we heard also yesterday, also from Martina Schüssler-Lenz from the CAT, the CAT chair, who also said that the CAT is very responsive and tries to help the developers also to work on such designs and to include the health technologies also way earlier than marketing applications, so to be able to address that earlier, and also their registries to get up registries running, which takes a lot of time, right? I would have another one for Jonathan, if I may.
Of course, please feel free.
You mentioned Dr. Google several times, so and that you had your family and friends searching also databases for you to find clinical trials. So would there be? I mean, as regulatory working with clinical trials, etc. If I'm looking for a trial, I'm looking at clinicaltrials.gov or EuropaBio or these kinds of databases mainly. So is it the databases you also looked at, or did it take quite some time to know that this is the database to look at? Or is it more really also other sources you have to check? Maybe also for maybe there are patients also listening for someone some advice.
Yeah, certainly. So I found I was pointed to both of those and looked at them. Both of them suffer a little bit from, they're fine if you know what you're looking for. Neither are, if it's the first time you walk up to them as a non-expert and you've never gone there before, the way the search engines work, you know, it's not always, you put mantle cell lymphoma and you get 500 hits, and it's not very helpful. And you need, once you've learned it, and it doesn't take long, it's fairly easy to navigate. But there's certainly, they're not. I think there's some usability there that could happen to make it easier. But in the end, so the thing about Dr. Google is that there's so much rubbish out there that is useless. There's so much very good stuff, but limited because of regulatory. You know, there's some wonderful cancer societies and great, but they're limited in what they can say because it has to be general. And of course, when it's about something new like CAR T-cell therapy or any of the ATMPs, there's a limited amount of information, and what they are allowed to release is limited. So actually, the most reliable source of information I found is patient to patient, and asking people for tips, who's talked to who, and you know, within a, for instance, funny, a silly one, when I was taking ibrutinib and having some fairly serious side effects, and I heard that acalabrutinib was similar efficacy but less side effects, I looked for a trial, but also discovered that it was being fast-tracked through the European Medical Agency. And so I found an email of a doctor who was on the committee, and I emailed him and said, hey, what about it? And obviously he couldn't say anything, but the letter I got back was just charming and wonderful and full of hope. It was wonderful. So, you know, when your life is at stake, you'll do anything. And I'm, you know, in this modern world, it's not that hard to find stuff if it's there. It's harder to know what to trust. And I think that's where the patient piece comes from. And of course, the clue, but I've had people come to me now who've been passed on, and I pass on the information. So it's a little bit of a mixture. I think you also have to be careful. Very well-meaning friends will tell you of the latest, latest fruit that was found in the back of the Amazon jungle. That is, you know, I know someone who was completely cured. There's all of that as well, and I think you have to be, you know, I think that's where my scientific background helps, being a little bit skeptical. But I'd say anything that anyone could do to help the patient-to-patient, peer-to-peer contact would be very helpful.
Thank you both.
Thank you, Jonathan, for answering the questions, and Annabelle and everybody for your participation, your time, your dedication through the entire process and the communication we had before the event itself, organizing it, discussing it. It was a real real pleasure for me to be in contact with you and to share this event with all of you today. Thank you once again. And I also have the honor today to close the event in general with three close messages from the Biotech team. First and most important, of course, is the thank you message. Thank you to all the speakers, the sponsors, the attendees for choosing to be part of Biotech Atelier. In general, yesterday and today, this is now established to be in yearly event and to be a European biotechnology conference that brings together scientists, business leaders, international experts from various fields, progressive thinkers, and people who want to exchange new idea about innovation, entrepreneurship, business development, technology, healthcare, life science, and pharma industry in general. So thank you all once again. The second, that is also very, very important message for us from the Biotech Atelier team is please save the date in 2021, because the year will grow even, the event that year will grow even bigger. It will be 3 days from the 29th of September till 1st of October. So I hope we all meet again next year. And don't forget to follow up, because all of the things that we have said, discussed, they're very, very valuable. Should be further developed, and staying in contact, exchanging ideas and information about progress is crucial. Thank you very much, everybody. I wish you a very nice rest of the day and looking forward to see you soon.
Thank you.
Thank you very much. Thank you.
Bye-bye.
Thank you.
Bye.
Thank you.
Bye.
Bye-bye.
Managing the development and life cycle of a pharmaceutical product can be beyond time-consuming, beyond costly, and beyond complex. PharmaLex enlightens with trusted expertise and empowers with cutting-edge technology. PharmaLex ensures healthier business decisions with specialized services that are adapted to fit your needs. Scalable yet approachable and innovative. Our services extend beyond market approval to include product maintenance and post-launch activities, both at a local and global level. PharmaLex, beyond knowledgeable, beyond flexible, beyond innovative, beyond trusted. And with over 90% of business from satisfied repeat customers, beyond proven. Let's take your business above and beyond. Contact us today.